So good afternoon, everyone. Am I audible to you all?
Yes, ma'am.
Yes, ma'am.
Yes,
Good
ma'am.
afternoon.
So I welcome the judges on the behalf of RAC. So we have with us today Dr. Anil Awad. We have with us today Dr. Sucheta Pulvarkar Madam from Biotech Department and Dr. Veena Kelkar from Biology, Botany Department.
Hello, everyone.
So welcome, Madam. So
Everyone.
Sucheta Madam and Veena Madam, with your due permission, should we start the sessions?
Yeah. Yes, ma'am.
Sure.
Dear students, just remember one thing, I'll be posting attendance. Make sure you're signing your attendance. Okay. So we will start with our session. The first session, the first presenter will be Mr. Omkar Yadav. Om Yadav. Om Yadav, you're here. So I'm making you co-host.
You want to share the attendance sheet first or at the end, ma'am?
Should I share in between?
Yeah, no problem. Or we can have it at the end also, not a problem.
We will have it at
So
the
that
end.
the students will also, yeah, so that the students will also wait for the others, you know.
Yeah, otherwise they will leave, most likely.
Yeah.
Okay. So should we begin?
Yes, ma'am.
Students, just you will be given 8 minutes for your presentation and 2 minutes will be question and answer session. So please try to finish in time. So Om Yadav, I have made you co-host.
Yes, ma'am.
Okay, so you can present.
Ma'am, may I start?
Yes, please start.
A very good afternoon to one and all. I am Om Yadav from class SYBSC. Today, I am going to present the research paper whose title is Virus Reconstitution and the Proof of the Existence of Genomic RNA, which was given by H. Frankel Cornet and B. Singer. The workplace of research was Virus Laboratory at University of California. The research work was published on January 20, 1956 in the Journal of the Royal Society. Dr. Hans Ludwig Frankel Cornet and Dr. Beatrix Braddon Singer discovered that RNA acts as genetic material. Dr. Frankel Cornet was awarded by Albert Lasker Award in the year 1958 for this discovery. Introduction. In this research work, tobacco mosaic virus TMV was used and the scientists demonstrated the landmark discovery that in some cases RNA acts as genetic material. TMV does not contain any DNA and it is made up of only RNA and protein molecules. They found that the virus could be broken into component parts and they could again be reassembled or reconstituted to form a functional virus. Their laboratory experiments proved that the nucleic acid could enter plant host cell and could generate identical new virus particles. This finding was the basis for the discovery of reverse transcriptase. Significance of the discovery. The experiment was the first evidence for the landmark discovery that RNA also has capacity to carry genetic information. This finding break the thinking that only DNA can be a genetic material and was a counterexample for the thinking that RNA don't have any biological significance. During the research, the scientists made world's first synthetic virus, and this discovery opens a whole new field for research activity. Tobacco mosaic virus is world's first discovered virus in the year 1892, and it has a rod-shaped structure. This is an electron micrograph of TMV virus. The virus causes tobacco mosaic disease in plants. This image
Om,
shows
can
an...
I
Yes, ma'am.
Can I just correct you here? It is the first plant virus to be discovered, bacha.
Yes, ma'am.
Yeah. So
Yes.
it's not the animal or the human virus. It is the first plant virus to be discovered.
Okay, ma'am. The
Yeah.
virus causes tobacco mosaic disease in plants. This image shows an healthy tobacco leaf and a leaf infected by TMV. Now, I am going to explain methodology combined with result for the better understanding of the experiment. TMV was degraded by treatment with detergent SDS, that is sodium dodecyl sulfate, and pH 10.5 buffer. Isolation of TMV in its native or renaturable form were obtained by using acetic acid. At pH 6, the mixture of these almost non-infective components, that is protein and RNA, show to reconstitute active viruses. Protein and nucleic acid fractions were isolated from different strains of TMV, like the wild type or common TMV, masked, Acuba, and the Holmes ribgrass strain. The plant used in experiment was Nicotiana tabacum, commonly known as tobacco plant. The experiment was divided in different sets. The set 1 of experiment is as follows. Separated RNA and protein molecules were used for infecting tobacco plant. When protein was inoculated on leaf, no infection was developed. But when RNA was inoculated, infection developed on leaf. The absence of contaminating viruses in RNA preparation could be proven by various methods. On ultracentrifugation, the infectivity of RNA was not sedimented, while viral infectivity was sedimented. Action of ribonuclease shows that RNA infectivity was extremely sensitive to ribonuclease, while TMV was quite unaffected by this enzyme. TMV antibody did not inactivate the infectivity of RNA, while TMV got inactivated. It was observed that virus rods, that is protein, was not causing infection in plant, but RNA alone was capable of causing infection. So it is regarded as the property of nucleic acid, that is RNA itself. In the second set of experiment, they selected two different strains of TMV. Those strains were disrupted and their RNA and protein were used. They created hybrid virus, also called as chimera virus, by using protein from TMV strain and RNA from HR strain. HR protein differs from TMV protein in amino acid composition and antigenic specificity. The HR protein contains methionine and histidine, which is absent in TMV protein. When this hybrid was treated with anti-TMV serum, its infectivity was largely neutralized, while anti-HR serum did not appear to affect it. Immunologically, therefore, the hybrid resembled TMV, the virus which has supplied the protein code, while its symptoms in the plant were only those of HR strain, which supplied the nucleic acid core. This hybrid, which was created by protein from TMV and RNA from HR strain, were injected in plants to obtain the progeny virus. The new viral progeny were identical to the strain from which RNA was isolated, that is HR strain. The progeny virus did not exhibit the property of strain which donated protein. The RNA in the virus determine the specificity of progeny protein. Conclusion of the original paper. The experiment concluded that genetic information controlling viral reproduction was contained in RNA at core of each virus particle. That is, RNA represented the genetic component of virus. Thus, the experiment demonstrates that RNA is the genetic material in TMV. Relevance to other discoveries. RNA code for synthesis of protein directly, while DNA depends on RNA. There is rapid rate of mutation in RNA molecules. During evolution, the RNA was first molecule to show the property of genetic material and biological catalyst both. RNA is essential in synthesis of polypeptide, ribosome, and ribonucleoprotein complexes. It is an essential in processes like transcription and translation. There was better understanding of virus reproduction, its spread in victims, its activity and diseases. The discovery of infectious nucleic acid provides a new approach to nature of reproduction of living things, to the problems of mutation and transformation of normal cell into cancer cell. These are my references for the books which I had referred and the photo credits. And thank you for listening to me. Thank you.
Thank you, Om. A very good presentation. Can you just go back to the HR? What was this HR strain?
Yes, ma'am. Ma'am, this one?
No, no, further. When that hybrid was made from one protein X or yeah.
Yes, ma'am.
What does this HR stand for?
Ma'am, it stands for Holmes ribgrass strain.
Okay. So they made a hybrid. Okay, fine. Okay. All right.
Okay.
Fine. I have no questions.
Okay. So any questions for Om?
Yeah, Om.
Yes, ma'am.
It is good to see that you are participating in different events and carrying out your passion regarding the subject. Just wanted to ask you, why did you choose this particular paper? Is there any specific reason?
Ma'am, RNA is a simple molecule when compared to DNA, and this research paper provides us that it was the first discovery in the field of molecular biology that RNA, a simple molecule, can also act as a genetic material for many viruses. And this experiment was there in my syllabus also in previous semester.
Acha. Okay, so that was always fascinating you, and that's the reason why you took up this particular paper.
Yes, ma'am.
Okay, so did it help while preparing for it to understand it better than what you thought it before?
Yes, ma'am. In syllabus, we came to know about only the significance of discovery, but while studying the research paper, we can understand the whole methodology and the whole procedure of experiment and what conclusion, how scientists derive conclusion, logical thinking, what they apply, all we came to know.
Okay, and did you back reference regarding the hurdles that they came across? Because there were a lot which was especially for this particular paper and the work that was done. So did you back reference for those experiments?
Ma'am, I have also referred to two papers which provide us with the methodology in detail.
Okay.
And I have mentioned its references also.
Very good. Okay, fine. Yeah. All the best, Om.
Thank you, ma'am.
Om, just tell me your
One question, Om. Okay, could you just tell me how did you search for this paper?
Ma'am, as I've studied in previous semester, I've typed it on Google, and I've searched for many references by searching it from Google.
And which language was this paper? Which language was this paper in?
Language?
कौन से language में था ये paper? Was it in English?
Yes, ma'am. It
Or
was
was it
in
in some
English.
other language?
No, it was in English
It
only.
was in English.
Yes,
Okay,
ma'am.
fine.
Om, you can just tell me your roll number.
Ma'am, it is 763.
Okay, thank you so much. Madam, any more questions from your side?
No, I'm good.
And fine.
Okay, thank you. Should we take the next participant?
Yeah.
Thank you, ma'am.
Okay, sure.
the
Next
best.
participant. Okay, thank you, Om. Can we have the next participant? The next participant is Muskan.
Sure.
One second. Our next participant is Muskan Khan from TY. Muskan, are you ready?
Yes, ma'am.
Okay, and after that it will be Diksha Yadav. So be ready, Diksha Yadav also. Oh, get la. Muskan, I have made you co-host.
Ma'am, is my screen is visible?
Yes, it
Yes.
is visible, Ms. Khan.
Yes.
Can I start, ma'am?
Yes.
Yes.
Good afternoon, everyone, and welcome to my presentation. Myself, Muskan Khan from TY BSc Zoology, roll number 630. As you can see on the screen, my topic is RNA-dependent DNA polymerase in virions of RNA tumor viruses. Two independent group of investigators, they are David Baltimore and Howard M. Tamin, have found evidence of an enzyme in virions of RNA tumor viruses which synthesize DNA from an RNA template. David Baltimore, born on March 7 in 1938 in New York, he is the virologist of US. He discovered an enzyme that synthesizes DNA from RNA. The enzyme reverse transcriptase has become an invaluable tool in recombinant DNA technology. He also conducted research that led to an understanding of the interaction between viruses and the genetic material of the cell. Introduction. DNA seems to have a critical role in multiplication and transforming ability of RNA tumor viruses. The discovery of reverse transcriptase has revolutionized modern molecular biology, and it has aided in the understanding of viruses like HIV and genetic basic of cancer. Reverse transcriptase is an enzyme and an RNA dependent DNA polymerase that was discovered in many retroviruses. Logic behind the study is no enzyme which synthesizes DNA from an RNA has been found in any type of the cell unless such an enzyme exists in uninfected cells. His study demonstrates that an RNA dependent DNA polymerase is present in two RNA tumor viruses, that is, roster mouse leukemia viruses RMLV and Rous sarcoma viruses, that is, RSV. Precedents exist for the occurrence of the nucleotide polymerases in the virions of animal viruses, a double-stranded RNA virus and a single-stranded vesicular stomatous viruses. Here are the methods, materials and methods. First one, properties of roster mouse leukemia virus. DNA polymerase. The reaction required magnesium, although manganese could partially substitute, and each of the 4-deoxyribonucleoside triphosphate was necessary for the activity. The reaction was stimulated strongly by dithiothritol and weakly by NaCl. None of these treatments increased the activity of RMLV, that is Roster mouse leukemia virus DNA polymerase. Second, characterization of the product. The product could be rendered acid soluble by either pancreatic deoxyribonuclease or micrococcal nuclease, but was unaffected by pancreatic ribonuclease or by alkaline hydrolyse. The product therefore has the product properties of DNA. Magnesium chloride enables the product to be digested by deoxyribonuclease. Effect of ribonuclease on the DNA polymerase activity of roster mouse leukemia virus. When 50%, when 50 microgram per milliliter of pancreatic ribonuclease was included in the reaction mixture, there was a 50% inhibition of activity during first hour and more than 80% inhibition during the second hour of incubation. Fourth one, the comparison of nucleotide incorporation by vesicular stomatis virus and roster mouse leukemia virus. The RMLV polymerase incorporated only deoxyribonucleotides at pH 8.3. Incorporation was readily demonstrable, but replacement of DATP and by ATP completely prevented synthesis. At pH 7.3, the RMLV polymerase was also inactive with ribonucleotides. The polymerase in RMLV variants is therefore highly specific for deoxyribonucleotides. Localization of the enzyme and its template. First one, to investigate whether the DNA polymerase and its template were associated with the virions. A RMLV suspension was centrifuged to equilibrium in a 15 to 50% sucrose gradient, and fraction of the gradient were assayed for DNA polymerase activity. The kinetics of incorporation of radioactivity from 3-http by RMLB are shown in curve 1. Most of the activity was found at the position of the visible band of virions. The arrow indicates the position of a sharp visible band of light scattering material which occurred at density 1.16 gram per centimeter cube. Here are the results and the discussions which we saw in materials and methods. First one, virions of the RNA tumor viruses contain RNA but no DNA. The formation of virions by the RNA tumor viruses is sensitive to actinomycin D and therefore seems to involve DNA dependent RNA synthesis. The template for the virion DNA polymerase is therefore probably the viral RNA. Pre-incubation, increasing the concentration of ribonuclease during pre-incubation could inhibit more than 95% of DNA polymerase activity. Deoxyribonucleotide incorporation measured in these experiments could be the result of an RNA polymerase activity in the virion, which can polymerize deoxyribonucleotides when they are provided in the reaction mixture. Here are the reference of my paper presentation. Thank you.
Muskan? Am I
Yes,
audible?
ma'am.
Yeah. Could you tell me a difference between a virion and a virus? Is there any difference or are they the same?
Yes, ma'am. I couldn't hear
What
you.
is it? Yeah. What is the difference between a virion and a virus?
Virions and the virus. Virions are free RNA molecules of low molecular weight,
Hmm.
and viruses can have
So,
either.
what does it lack then? That if you're saying it's free RNA molecule, what is it lacking? Does it lack something?
Yes, miss, without any protein coat.
Okay, all right. And what is this pre-incubation which you have mentioned? What is in the, I think in the second last slide you've spoken about pre-incubation.
Miss, prior to the treatment or the process.
Yeah. So where is this pre-incubation done?
Ma'am, before the
And how
centrifugation in the lab.
sorry?
Before the centrifugation in the lab.
Yeah. Does it have any specific temperature or specific condition? And what is its
Ma'am,
advantage? What was the need to do that?
it has, ma'am, it is done in room temperature.
Okay.
Advantages.
All right.
Mm.
Yeah. And what is? Could you just show me the first slide? I want to know how is this discovery help in the
Yes,
recent
ma'am.
future, or yeah, give me few of its applications. Okay, where is it being used? Yeah.
Ma'am, reverse transcriptase are used by viruses such as HIV and hepatitis B to replicate their genomes by retroposons, mobile genetic elements.
Okay. Fine. Fine. Go back to the first slide, beta.
Right. Folder है ना?
इसको भी tell. इसको भी tell. The first slide.
आणि
Ma'am,
if
this is the first slide.
Yeah. Yeah. Fine. Fine. See. And this paper was published in which year?
Ma'am, in 1970.
And in which journal? 70. And which journal?
Nature volume 226.
Oh, fine. Thank you so much.
Thank you, ma'am.
Yeah, Muskan.
Yes, ma'am.
Yeah. So, what is the significance of this particular discovery in virology?
Ma'am, it is needed for the function in viruses, in eukaryotes and prokaryotes. The enzymes in the cells are involved in genetic diversity.
Okay. And what is David Baltimore's contribution in virology? Do you know?
Ma'am, he discovered the enzyme that synthesized DNA from RNA.
Anything regarding the classification? About which you're talking, you had selected the paper. So anything you know regarding the classification part and David Baltimore and virology?
Ma'am, he described that no enzyme which synthesizes DNA from RNA template has been found in any type of cell.
Yeah, okay, fine. I mean, not the work, I'm talking about the classification. Okay, fine. That's okay.
Ma'am is giving you hint. Talking about classification, she's directly giving you a hint.
Yeah. Because we have been hearing about this. Even Sir talked about his contribution. Obviously, his contribution is immense, and of the Nobel Prize winner when you talk about. So what I would want each and every one of you to have some kind of back reference. Okay, so develop that habit. Know more about it so that you will enjoy more and you will learn more. All right.
Thank you, ma'am.
Yeah. Okay. Fine.
Thank you, ma'am.
Okay, thank you, judges. Thank you, Muskan. Madam, with your due permission, can we have the next participant?
Yes. Yes. Sure.
Yeah, thank you. Can we have the next
Ma'am,
participant?
you need not take our permission. Please go ahead. It is very embarrassing for us.
And then what?
Okay. Can we have the next participant? I think Diksha Yadav.
Yes, ma'am.
Okay, Diksha, I have already made you co-host.
Is my screen visible, ma'am?
Yes,
Yes, Bachcha. Yeah.
Hello.
yes, Diksha.
Yes.
Please go ahead.
May I start?
Yes.
Yes, please.
Sure.
Good afternoon, one and all. Diksha Dinesh Yadav of class SY Division C. Today, I am going to present Temmin's and
Diksha,
Satoshi's research paper on
sir.
RNA dependent.
दीक्षा बेटा जस्ट मोमेंट हाँ अभी शुरू हो गया तुम्हारा प्रेजेंट
हाँ.
हाँ अभी या नाउ
वापस
यू
बोलूं, मैम?
हाँ शुरू करो अभी अभी दिख रहा है प्रेजेंट
Okay, ma'am. Good afternoon, one and all. I am Diksha Dinesh Yadav of class SYBSC Division C. Today, I am going to present Temmin's and Satoshi's research paper on RNA-dependent DNA polymerase in Virion's years volume 226 on 27 1970. Howard Temmin was American geneticist and virologist. He got Nobel Prize in 1975. He did his research work at McArdle Laboratory for Cancer Research at University of Wisconsin. He discovered reverse transcriptase in the year 1970. Nobel Prize in Physiology or Medicine with Renato Del Becco and David Baltimore, as they discovered the interaction between tumor viruses and the genetic material of this cell. Now, what is reverse transcriptase? Reverse transcriptase, also called as RNA-directed DNA polymerase, an enzyme which was encoded from the genetic material of retroviruses that catalyzes the transcription of retrovirus RNA into DNA. This catalyzed transcription is the reverse process of normal cellular transcription of DNA into RNA, hence the name reverse transcriptase. This is landmark discovery because the virus with genome consisting of RNA can also be inserted into host cell DNA. We are talking about transcription and reverse transcription processes here. Let us understand this first. What are these processes so that we can get a clear idea about it? When DNA template is used to make RNA, the process is called as transcription. Transcription occurs in prokaryotes and eukaryotes. In prokaryotes, it mainly occurs in cytoplasm, while in eukaryotes, transcription occurs in nucleus. In transcription, RNA polymerase is involved. The product of transcription are used in protein synthesis, while when RNA template is used to make complementary DNA, the process is called as reverse transcription. It mainly occurs in retroviruses. Reverse transcription occurs in the cytoplasm of the host. In reverse transcription, reverse transcriptase is involved. The product of reverse transcription get integrated into the host cell DNA. These are the pictorial representation of transcription and reverse transcription processes respectively. To understand what brought changes in RNA tumor viruses, scientists designed number of experiments to understand this action in Rao sarcoma virus. In first set of experiment, The virons were purified. The method used was virus was centrifuged in 15% sucrose cushion and later purified by equilibrium sucrose density gradient centrifugation technique, which resulted into pure stock of virons. Now, in second set, activation of enzyme was carried out. Virons were subjected to non-ionic detergent DTT, resulting into disruption of virons of RNA tumor viruses. In third set, requirement of enzyme activity was seen. MnCl2 is substituted with MgCl2 in polymerase assay, but not with CaCl2. This result of this experiment was the amount of disrupted virons present was varied and the amount of incorporation varied with the second order kinetics. In fourth set, to obtain the source of polymerase, virons of SRV and AMV subject to nonidot. It is a radio label, resulting in normal cell supernatant with no activity. But when disrupted virons of SRV and AMV were subjected to nonidot, virons of AMV was obtained with 10-fold increased activity. In fifth set, polymerase activity was examined. Method used was disrupted virons and ribonuclease A were reacted at 80 degrees Celsius for 10 minutes. Various results were obtained. For full activity, all 4-deoxyribonucleotide triphosphate were required. But when 3-deoxyribonucleotide were present, some activity was still present. When 2-deoxyribonucleotide was present, 10 to 20% of the full activity was seen. But the main important discovery of this experiment was intact RNA is necessary DTT. Nature of products are
Diksha, you are not audible. I think there was some network issue.
Yeah, there was quite a lot of disturbance, I guess.
Diksha?
Hello.
Diksha,
Yeah.
Hello.
yes.
Yes, ma'am.
Your screen is not visible now.
Yes, ma'am. 1 second.
Yeah.
Ma'am, from where should I start now? Diagrams and charts, should I show this?
Yeah, show that.
Hello.
Yes.
Yes. For these are the diagrams and charts which I took from original paper for activation of enzyme, polymerase activity, MgCl2 requirement, that is an enzyme requirement, source of polymerase, nature of the product. Now, conclusion in this paper, there is a new polymerase inside virons of RNA tumor virus. In normal cell supernatant, polymerase was not present, while in virons of avian sarcoma and leukemia RNA tumor virus, polymerase was present. Significance of this discovery is that it resulted into identification into new unique virus family, that is Retroviridae, and the understanding of pathogenesis of these viruses. Now, relevance of this discovery in today's time. It is the counter example of central dogma. This experiment also proved DNA pro-virus hypothesis. It has a great influence on cancer biology. Also, the RT-PCR technique used nowadays is also based on the activity of reverse transcriptase enzyme. These are the references and photo credits I used for this presentation. Thank you.
Yeah, thank you, Diksha. Could you give me one example of retrovirus? You mentioned that.
Yes, ma'am. HIV.
Yeah.
HIV is a retrovirus, and Rous sarcoma virus, which Temin used, is also a retrovirus.
Yeah.
HTLMV is also a retrovirus.
And all retroviruses have RNA as its genetic material?
Yes, ma'am. Ma'am, actually, they are retrovirus because they use reverse transcriptase enzyme. That's why their name is retrovirus.
Okay, and what is that intermediate product that is formed? It has its template as your RNA, right?
Yes, ma'am.
Okay, so after
It has.
Yeah, go look.
Yes, ma'am. It has RNA template, which is used to make DNA. The intermediate which is formed is hybrid, where one strand is RNA and the second strand is DNA.
So if this is also DNA and normal is also DNA, तो उससे कोई फर्क है कि नहीं है?
Yes, ma'am. फर्क है जब भी ये वाला जो RNA का टेम्पलेट यूज कर रहे हैं और DNA बना रहे हैं.
हाँ.
हाँ, तब हम लोगों को एक स्ट्रैंड ऑफ DNA मिल गया. उसके बाद में ना एक एंजाइम यूज करते हैं ताकि वो जो RNA टेम्पलेट है उसको डिसॉल्व कर देंगे और उसके ऊपर हम लोग जो DNA हम लोग का अभी बना है उसके ऊपर दूसरा होगा और फिर अब हमारा डबल स्टैंडर्ड DNA बनेगा.
Okay, Diksha.
Yes, ma'am.
Yeah, so suppose that you were in this particular group, okay, doing the research. Okay.
Yes, ma'am.
So what could be one thing that you wouldn't have done, and what could be one thing that you could have done?
Ma'am, actually, I think all these experiments have their significant role. I couldn't delete anything from this.
Yeah, okay. So what could you add more?
Ma'am, maybe I will do in simple language.
Oh. Okay. And is there anything why you chose this particular paper?
Yes, ma'am. In last semester, we had this in our portion. So that's why fascinating laga.
Acha. So did it help you to understand it better?
Yes, ma'am.
Okay, so is there anything what you had not understood before and what this paper made you understand?
Yes, ma'am. पहले नहीं पता था different how they scientists work, मतलब how they design different experiment. ये सब नहीं पता था. But after this, I got to know.
Right. Okay. Fine.
Okay, then should we take the next participant?
Yes, yes.
Okay, next participant is Shilpa Shukla. And after Shilpa Shukla, it is Kiran Vishwakarma. So Kiran, you also be ready.
Yes, ma'am.
Shilpa, you can start.
Shilpa? Shilpa, are you there?
Sorry.
Her share audio is off.
Ma'am, I was disconnected. Can you make me co-host again?
Okay. Okay, Shilpa, I have made you co-host.
Ma'am, is my screen visible? Hello.
Yes, yes,
Yes,
yes.
yes,
May I start, ma'am?
yes,
Yes.
please.
Good afternoon, everyone. I am Shilpa Shukla, studying in SYBSc from Ram Ranjan Jhunjhunwala College. I am going to present a research paper whose title is Glycogen Formation in the Liver from D and L Lactic Acid, which was given by Carl F. Corey and Gertie T. Corey. They both were American biochemists. They did their research work at the State Institute for the Study of Malignant Disease, Buffalo, New York. They discovered Cori cycle in the year 1929. They shared Nobel Prize in the Physiology or Medicine in the year 1947 with Argentine physiologist Bernardo Hauser. Gertie Cori was the first American woman to win Nobel Prize in Physiology or Medicine. They also discovered Cori Easter. This is a picture of original paper. Now, what is a Cori cycle? Cori cycle is a cyclic process in which lactate produced by the skeletal muscles during anaerobic respiration is transported to liver and converted to glucose, which is cycled back to the muscles. It is also known as lactic acid cycle. As we all know, under aerobic condition, glucose is converted into pyruvate through glycolysis, and pyruvate goes in TCA cycle and led to the formation of NADH and FADH2. In anaerobic condition, no TCA cycle will occur after pyruvate formation as there is no oxygen, which is terminal electron acceptor. Therefore, ETC cycle will not occur. Therefore, formed pyruvate is converted into lactate by enzyme lactate dehydrogenase. Lactate is formed in the RBCs and the muscles during anaerobic respiration. Do you know our RBCs lack mitochondria and therefore they rely on anaerobic metabolism for ATP. As lactate is dead end of glycolysis and it has to be converted to pyruvate for the further metabolism. Lactate produced in the skeletal muscles cannot be used for the production of glucose due to lack of enzyme glucose 6-phosphatase and fructose 1,6 bisphosphatase, which is required for gluconeogenesis. The plasma membrane is freely permeable to lactate. Lactate is converted from skeletal muscles through blood and handed over to liver, where it is oxidized to pyruvate. Pyruvate so produced is converted to glucose by gluconeogenesis, which is then transported to skeletal muscle. This is a cyclic representation of Cori cycle. Vigorous exercise in muscle can lead to oxygen shortage and energy requirement must be met by increased level of glycolysis. Under these conditions, glucose breaks down into lactate, but this could lead to an intracellular accumulation of lactate and a consequent reduction in the intracellular pH. This cycle prevents lactic acidosis. For proving this cycle, this Coreys perform many sets of experiments, out of which I am highlighting major 3 experiments. In the first set of experiment, they measure the glycogen content of 24-hour fasting rat. And in another set of experiment, they feed lactic acid via 2 modes, one by subcutaneous injection, which utilizes slowly and works as a muscle lactate, and one orally. After 3 hours of injection, they measured the glycogen content of liver. uh, these are the observation table which was given in the paper. Now, what was the topic of discussion to the scientists? On comparing the data of table 1 and table 2, they found that liver is able to form glycogen from lactic acid. After that, they used different isomers of lactate for comparative studies. They found some results like blood sugar level hardly changed during lactic acid absorption. Isomeric sugars are absorbed at widely different rates from intestine. For example, mannose is absorbed 5 times more slowly than glucose, but D and L lactate are absorbed at nearly same rate. L lactate is able to form liver glycogen at a very slow rate. Now, let us move towards the conclusion of this research. Sodium D-lactate and sodium L-lactate are absorbed at the same rate from the intestine, but sodium L-lactate forms hardly any glycogen. L-lactic acid is utilized 4 times more slowly in the rat than D-lactic acid. This is a cycle which was given by Coris in the original paper. After that, they studied effects of hormone on the cycle. Firstly, they studied effect of epinephrine. Epinephrine accelerates the cycle in the direction of muscle glycogen to liver glycogen and results in accumulation of sugar in the blood. After that, they studied effect of insulin. Insulin accelerates the cycle in the direction of blood glucose to muscle glycogen, which led to hypoglycemia and secondarily to depletion of glucose stores of the liver. Now, what are the significance of these landmark discoveries? It connects gluconeogenesis with anaerobic glycolysis. Cori cycle prevents lactic acidosis, that is excessive accumulation of lactate in the muscles under anaerobic conditions. This cycle is also important for the production of energy molecule ATP during muscular activity. It helps in proper functioning of muscles by supplying required energy in the form of glucose. Cori cycle is a much more important source of substrate for gluconeogenesis than food. From this discovery, we can understand that the glucose molecule is able to complete its cycle in the body. This discovery led to an improvement of techniques to control diabetes. These are the some references and the photo credits which I used for the presentation. Thank you.
Shilpa, can you give one disadvantage or say drawback of lactate in the body, accumulation of lactate in the body?
Ma'am, accumulation of lactate leads to change in the pH of blood. As we all know, our entire body is dependent on metabolism, so which require enzymes. So slightly change in pH can cause effects of enzyme and our metabolism will be stopped.
And what about in the muscle?
Ma'am, same as muscles during anaerobic respiration, uh, glycolysis occurs at the same. After that, glucose enters. After that, the pyruvate which is formed enters the lactic acid cycle, converted into the lactate. Then muscle cannot use these lactate as they lack the enzymes, which I mentioned, glucose 6-phosphatase and fructose 1,6 phosphatase. So these are transported into the liver.
All right, Shilpa,
Yes, ma'am.
do you know anything that could be enhancer or stimulator of Cori cycle?
Sorry, ma'am. Can you repeat?
Any enhancer, regulator of Cori cycle?
Ma'am, like as we said, Cori cycle depends on as muscle glycogen is converted into the liver glycogen.
Right.
Epinephrine is an hormone which
Right.
accelerates this cycle.
Right. Okay. So what happens? How does it stimulate epinephrine? How does it stimulate?
Ma'am, as epinephrine is known as 3F hormone, so it accelerates the metabolism, like utilization of the glucose. So it led to and more utilization of the glucose in a shorter time led to the oxygen the shortage. So therefore, it converts into lactate. So muscle glycogen is broken down
Right.
into the lactate, and this lactate is converted into the liver with the help of these cycles.
Right. So breaking down of glycogen is
Yes, ma'am.
what is it called as that particular process?
Breaking down of glycogen, glycogenolysis.
Okay.
Glycogen lysis.
Right. Okay. Very good. Okay. Shilpa.
Thank you, ma'am.
So can we have the next participant then?
Yes, yes.
Kiran,
Yes, ma'am.
you are the next one. And after Kiran, I have made a co-host to, so you also be ready. Okay, Kiran, you can start. Students have posted the attendance link, and the teachers who are present, Sanika madam and Sushma madam, I welcome you all also for the function. Madam, please do sign the attendance sheet.
Ma'am, can I start?
Yes.
please start.
Good afternoon, everyone. Today, my topic for presentation is selection of high nicotine producing cell lines of tobacco callus by single cell cloning, which is given by the Taiko Ogino, Noburo Hirauka, and Momuro Tabaka from Faculty of Pharmaceutical Science and Kyoto University, Sakyoku, Kyoto 606, Japan, Mitsui Petrochemical Industry Limited, Research Center, Wagi, Kugagun, Yamaguchi, 740, Japan, which is received in 7 April 1978. The structure of research paper is abstract, introduction, methodology, results, discussion, experiment, and references. The keyword index: Nicotina tabacum, Solanaceae tobacco, plant tissue culture, selection, variation, single cell cloning, and synthesis of nicotine. Abstract: the single cell clones of Nicotina tabacum callus showed wide variation in the production of nicotine, which is an efficient screening of numerous clones was made possible by the adoption of cell squash method for estimating the approximate alkaloid content of a small piece of callus. The clones, several cell lines with higher yield of nicotine, that is 1 to 3.4% of dry weight, has been isolated by the repeated clonal selection. Introduction. Davison described the presence of nicotine in the tobacco callus in 1916. The several co-workers have reported the synthesis of nicotine by the various callus culture of Nicotina species. The nicotine content of these callus culture was slightly low compared with those plants, except for the culture reported by the Ofta et al., which show content as high as 1.7% of the dry tissue. Second, the plant growth regulator and shows wide variation in the producing capability among single cell clones isolated from a callus tissue of Nicotina rustica. There is a simple device for detecting clones with higher alkaloid is also described in this paper. Results: first, screening of clone by cell squash method. Second, isolation of high nicotine producing cell lines. Third, stability of the cell lines, time course of nicotine production. So first, screening of clones by cell squash method. It is the correlation between the nicotine content of single cell and clone and the score obtained by cell squash method. The score of each point present the mean of 5 tissue sample taken from each clone. As the primary screening test for a single cell clone with a higher yield of alkaloid was carried out by utilizing the cell squash method, there the clone assays were scored from 0 to 4 according to the intensity of colored spots appear on the filter paper underneath the tissue. The actual nicotine content varied to the considerable extent even among the tissue with the same score, as high correlation coefficient are, that is mean 0.80, was obtained between the value measured by two different methods, which is suggested by the cell squash method. Second, frequency of distribution of single cell clones with different nicotine content. Clones isolated first, cloning of original callus culture, and second, clone developed from single cell of a parent clone which shows the highest nicotine content in the first clonal selection. In the process of selection, only those clones with higher scores were under-cultured to provide sufficient amount of callus tissue necessary for confirming their actual nicotine
content by UV. The isolating of high nicotine producing cell lines. The graph shows effect of repeated selection of nicotine content in various cell clones. Single cell clones from the original strains with high nicotine content was 0.7%, showed wide variation in the nicotine content. The score obtained by the cell squash method varies to shown in the graph, that is 1, 2 or 3. The majority of clones scoring 1 and 2 have the highest nicotine content. Such variation in the nicotine content was observed also in a single cell clones of the second cycle, which were isolated from a callus culture, showing the highest nicotine content in the first clonal selection. The greater the number of clones of the second cycle obtained more nicotine than the first cycle, as shown in this figure. Third, stability of the cell lines. Fluctuation in the nicotine content of 5 selected cell lines during successive generation in the absence of artificial selection for nicotine content. Each arrow indicates the date of isolation of the high nicotine producing cell lines. The stability of the cell line, as shown in the figure, that is 772, T142, T14, T57 and T49. During a long subculturing, their nicotine content were measured at consequence transfer generations. The nicotine content of a tested line more or less fluctuated through a year of a subculturing without selection. The nicotine content of T72 once increased from 2.2 to 1 and then increased up to 2%. Like in case of T49, the nicotine content increased from 2.1 to 3.4% after 7th month. Next, the time course of nicotine production. There is the two graphs. First is the culture grown in the laboratory and the original strain. The time course of growth and nicotine production in the high nicotine producing lines in strain T14 and the original strain B. As shown in the figure, the time course of the growth and the nicotine production in the original strain and the selected line T14, both the cultures show similar growth pattern with the significant differences in the dry weight of tissue. The nicotine synthesis is initiated at the exponential growth phase in the both culture. The selected lines show higher rate of nicotine production during the growth period, but at the 6th week, the nicotine content and the dry weight reach maximum in both culture, but they gradually decreases as the tissue being deteriorated or at the stationary phase. It also concluded therefore that the high nicotine content of T14 is not due to the shortening of the lag period prior to nicotine synthesis, but it is due to the higher production of rate. Next is the discussion. There is many discussion on this, but most of the selection experiments reported dealt with the easily detectable variation in the pigments such as anthocyanin, carotenoid, and naphthoquinone, except for few work on the non-pigmented product. It is expected that this economical method could be applied to detection of not only the dragon drop positive alkaloid but also a variety of other compounds which react with the specific reagent which give color spots. It is also suggested by the studies on the single cell clones which is derived from the Nicotina rustica and Catharanthus roseus, that the callus culture consists of heterogeneous cells which differ in their production of alkaloids, and that selection of single cells are highly effective in increasing the alkaloid content of a single of the cell lines, which increases the quantity of serpentine and agemalacin up to 1.3% of dry weight in the selected cell line. The present study indicates the importance of isolation and selection of variant cell for establishing a superior cell line with a higher yield of successful secondary metabolites. Next is the experiment, which is in 4 parts. First is the culture stock and culture method. It is in this the callus culture stock, that is the which strain is used, the BY12H, the originally derived from the stem pith of Nicotina tabacum, that is tobacco, which is bright yellow, which is maintained in the Lismer's cook basal agar medium and supplemented with the auxin and kinetin at 25 degrees Celsius in the dark of 11 years by the successive transfer carried out within the interval of 1 month. Second is single cell cloning. The small piece of callus tissue were inoculated in the Lismer's cook liquid medium and supplemented with the kinetin in the Erlenmeyer flask, and which is kept on the shaker for 1 week, and the auxin is omitted from the medium because the dividing cells show a tendency to form a multicellular chain in the presence of auxin. So the auxin is omitted and the cell suspension obtained were filtered through the nylon sieve. The filtrate contained single cell and a small number of linear dividing cells consisting of 2 to 8 cells, which were too small to be removed by filtering. The filter are centrifuged at 80 rpm to concentrate the cells. The single cells of BY12H to grow on the flatted directly on the nutrient agar medium, so they are grown on the Lesmier cook medium. Third is the cell squash method. The primary screening of higher alkaloid producing clones were carried out by the cell squash method, which is in which the small piece of callus was sampled aseptically from each clone in the 5 pieces, which is arranged on the filter paper and which is pressed by the glass plate, so that each and every cells of the callus are underneath the filter, and then on the filter paper the dragon drop reagents are sprayed, so the alkaloid is visualized, which was scored according to the intensity of the color. When there is the alkaloid present, then the dragon drop reagent shows the color changes, that is from orange to orange red. The intensity of scores are on the basis of 0, 1, 2, 3, and 4. 0 is uncolored, 1 is faint, 2 is moderate, and 3 is deep, and 4 is very deep. When the clone scoring 3 or 4, then there is a higher alkaloid content in these cells. And a fourth, analysis of alkaloid, which is analyzed by the alkaloid by TLC and estimated by the nicotine content of callus tissue by UV. These are my references. Thank you very much.
Kiran, how do you estimate nicotine? You did single clone cell cloning, right, to
Yes.
enhance the production of nicotine from the nicotiana plant or callus.
Yes, ma'am.
Correct. What
Yes.
was the method for estimating the amount of nicotine?
Ma'am, by cell squash method.
Sorry?
Ma'am, by cell squash method.
What was that?
Ma'am, in cell squash method, the dragon drop reagent is spray on the cell, which shows the color like orange to orange red.
Which color?
Orange to orange red.
So nicotine gives color by spraying with the reagent.
Ma'am, alkaloid gives color. Nicotine is also, ma'am, a type of alkaloid. So
Okay. Okay. Fine.
Kiran, can you go back to that particular graph that you had shown for Catharanthus and Nicotiana?
yes, ma'am.
Students, I request, do not shut down your presentations till we finish with the questions. Okay, because sometimes we need to go back and forth with the presentation. So leave the presentation there. Once we are done, then you can shut down.
Yeah, time course. Yeah.
Yes, ma'am.
Yeah, so can you elaborate on this?
Yes, ma'am. In this graph or figure shows with the original strain and the T14 strain, which are grown on the same medium, which shows the significant difference in the dry weight of tissue.
Which medium was taken?
What, ma'am?
Which medium was taken?
Linsmer's Cook medium.
Okay.
Linsmer's. Yes.
Yeah, go on.
Then the nicotine synthesis is initiated as the exponential growth phase. In the second week, from the subculturing in the both culture. Then the selected line shows the higher rate of nicotine production during the growth phase of period.
Okay, can you cut or delete whatever? Yeah. And this is the maximum size that can be shown?
Yes, ma'am. Yes, ma'am.
Okay.
Oh, yes, ma'am. And then the nicotine content and the dry weight reach the maximum in both the culture as in original strain and in the T14. Then there is the gradual decrease in the tissue because due to there is the stationary phase and the cells or tissue being deteriorate. And therefore this concluded that the high nicotine content of T14 is not due to the shortening of the lag period prior to the nicotine synthesis, but it is due to the higher production rate.
Okay, that is what I was thinking. What was there any justification for that particular result or that particular inference?
Yes, ma'am. Yes.
Don't worry, don't panic. You can think and then you can answer, okay?
Yes, ma'am. It is concluded that the nicotine content of T14 is not due to the shortening of lag phase, but it is due to the higher production rate.
But is there any justification for that? Why do you think so?
No, ma'am. There is no justification. No, ma'am. No, as such, no.
So when you say so it can be concluded, that means there was something and hence it was concluded, right? So that's the reason I asked. Okay, fine.
Okay, ma'am.
Okay, yes.
Okay, ma'am.
So we have next one is Kalamuddin. After that there is Maqsood. So Maqsood, you also be ready.
Okay, ma'am.
Okay. I have already made you co-host, Kalamuddin, so you can please start with your presentation. We have Kiran sir with us and Dr. Geeta Joshi also.
Hello.
Hi. Who is the next speaker? Janvi ma'am?
Kalamuddin. Kalamuddin.
Kalamuddin. Okay.
Kalamuddin, are you there? I have already made you co-host. Kalamuddin Sheikh.
Yes, ma'am. Yes, ma'am. मैं शेयर कर रहा हूँ.
हाँ.
उसके बाद मकसूद अली है. So Maqsood Ali, you also be ready.
Yes, ma'am. Yes, ma'am.
Okay, we have our teachers with us. Dr. Vice Principal Dr. Kiran Kolvankar. Sir, we welcome you to the session. We also have head of the zoology department, Dr. Pratibha Sardesai. Madam, we welcome you.
Hello, thank you.
Geeta Joshi Madam is also there from zoology department.
Ma'am, can I start?
Yeah, sure, please.
Good afternoon to everyone. Today's my topic of representation is refinement of the triphenyl tetrazolium chloride method of determining cold injury. Work is done by Peter L. Stepankas and F.O. Lamphere. Workplace is Department of Horticulture, Purdue University, Lafayette, Indiana, 47906, received in June 21, 1967. Now first we see hardening. Hardening is the process by which a plant or tissue is made more resistant to any environmental extreme such as a low temperature. Now we see cold hardiness. The cold hardiness is the ability to resist injury during exposure to low temperature. Cold hardiness is a reliable method to determine tissue viability. Ideally, the method should eliminate bias associated with visual observation and be based on a quantitative system that could be analyzed statically and utilize a small quantity of a tissue, be relatively quick and capable of predicting the future performance of the plant. Primarily, TTC test has been used in the past as a quantitative response, and if the red formation derivative was produced, the tissue was considered to be viable. However, only limited provision has been made to distinguish between the varying degree of a TTC. As a result, difference has been reported: dark pink, pale pink, streak pink, or a pink. Another serious disadvantage of a TTC test that even if the sample of a tissue reduces the TTC to be formation derivative, the plant may be die the next day. Therefore, any refinement of the TTC test must be enabled to ascertain if the tissue sample was viable at the time of testing, will be indicative of a future viability of the plant from which they were taken. Now materials. Investigation in the refinement of a TTC test utilize the both leaf disc and stem segment of a Hydra helix linear, also called Thornden English EV plant grown in a greenhouse. Here designated plant were artificially cold acclimated at a 5 degrees Celsius for 6 weeks under the 600 feet candle of light provided by incandescent and cool white fluorescent lamp. Leaf and stem sample were frozen under control condition with the declined temperature for 3.3 degrees Celsius per hour. Now method. Procedure for the refinement of TTC is as follows: where the sample which is previously frozen, and the section of the stem section are cut to maximum length of 1 centimeter and 0.7 centimeter leaf disc. Place the tissue into 17 into 120 mm test tube. Make up the volume 10 ml and add 3.0 ml of a 0.6% TTC in 0.05 molar buffer and 0.05 wetting agent, also called Ortho X77, and infiltrate under the vacuum and incubate at the 30 degrees Celsius for 15 hours. Drain the TTC solution and rinse the tissue once with distilled water. Extract the sample with 7 ml of 95% ethanol in a boiling water bath. A 5-minute extraction period is adequate for the leaf sample, while woody tissue must be first be sliced and then extracted for the 10 minutes. 98% of formazan was recovered in the initial extraction. The extraction are cooled and made up of to 10 ml volume with 95% ethanol. Record the absorbance at 530 nanometer. This graph shows that that 95% of TTC, 95% of formazan are extracted by using the 98% of formazan was extracted by using 95% of ethanol from leaf disc and stem segment, which is incubated for the 15 hours for 30 degrees Celsius. Now, result and discussion. To facilitate the interpretation of the data and to compare result of a different experiment, the amount of formazan produced by tissue sample exposed to the freezing temperature was expressed as a percentage of the amount of formazan produced by unfrozen control sample. Result of a typical freezing test are presented. The amount of formazan produced rapidly declined after the temperature of -4 is reached, but the tissue of a cutting exposed to -7 is still able to reduce the TTC of a formazan derivative. Now, individual plant of a varying degree of a cold hardiness were separated into two leaf cutting and subjected to the temperature. The stem were assessed for their ability to reduce TTC. The remaining portion of the cutting was placed into the moist vermiculite at 21 degrees Celsius, and survival of the stem and leaf portion was usually determined after the 4 weeks. The percentage reduction was plotted with respect to the freezing temperature. The percentage reduction was represented with a dot if the corresponding leaf or stem segment was alive after the 4-week period, and the circle was if a circle was used if it was dead. Cutting sample of which had a 50% reduction value or greater were alive after the 4-week of a period. Conversely, sample which had a percent reduction value of a 50 or less were viable at the time of assay, but the cutting died during the following 4 weeks. The minimum amount of TCC reduction by tissue samples required to ensure the survival of the cutting at the later date is a 50%. And the killing point of the cutting is defined as a temperature where 50% reduction value is reached. Now, based on the definition, killing point of a tissue -6.50 for stem tissue and -8.1 for the leaf tissue. This graph shows that tissue which has a 50% reduction of a TTC are alive after the 4 weeks, and the tissue which has a reduction of a TTC less than 50% are dead after the 4 weeks. Now, this is the absorption spectrum.
Just a reminder, you have crossed 7 minutes. Just a reminder, time is up to 8 minutes only.
Okay, ma'am. Reduced TTC show maximum absorption as compared to the leaf extract plus leaf extract plus TTC and leaf extract only. The decrease in the TTC reduction with cold injury may be due to cold inactivation of dehydrogenase. As the dehydrogenase activity is difficult to demonstrate the previously frozen tissue. However, demonstration of a specific dehydrogenase by tetrazolium in a fresh tissue is difficult due to non-specific tetrazolium reduction. Freezing to allow the substrate to diffuse from the cell and subsequent addition of a specific substrate and cofactor makes the possible demonstration of a specific dehydrogenase. The previously frozen section required addition of a substrate and cofactor for intense staining, and section stain weak without the cofactor and not at all without substrate. Thus, high correlation between cold injury and TTC reduction system, where exogenous substrate and cofactor are not added, is due to the cold inactivation of dehydrogen, but due to the cofactor and substrate limitation. This limitation could be caused by inactivation of enzyme other than dehydrogenase required for the continuous synthesis of substrate and cofactor.
You have crossed 9 minutes.
Ma'am, is last slide.
Students, please stick to your time of 8 minutes.
The irreversible inactivation of oxidation phosphorylation by freezing represents such key step leading to depletion of a substrate and cofactor and subsequent decrease in the amount of TTC reduction in a manner similar to inhibition of a TTC reduction by 2,6-dinitrophenol. Substrate and cofactor limitation may also arise due to diffusion or dilution of a substrate. These are my references. Thank you.
Questions for him, judges, please.
Go back to the original first page, beta. First slide.
Okay, ma'am. Ma'am, this one?
Yeah, one second. Okay. Was this paper in English?
Yes, ma'am.
Okay. Okay, next slide. Fine, I have no questions.
Yeah, okay, Kalam,
Yes, ma'am.
just a basic question.
Yes,
Is
ma'am.
this TTC and formation of formazon, is it cytotoxic?
No, ma'am. एक्चुअली मैम हम लोग ना इससे कोल्ड इंजरी का मतलब पता लगाते हैं अगर टीटीसी को प्लांट जो भी हम लोग सेल और टिश्यू यूज कर रहे हैं वो टीटीसी को 50% रिड्यूस कर रहा है तो फिर वो वायबल होगा अगर टिश्यू 50% रिड्यूस नहीं कर रहा है तो आफ्टर सम टाइम तो मतलब कट करने के बाद वो डेड हो जाएगा, उसकी डेथ हो जाएगी।
किसकी?
मैम टिश्यू और जो मतलब जो हम लोग सैंपल मटेरियल यूज कर रहे हैं।
Yeah, right. So, no, what I meant to ask is that particular process cytotoxic?
No, ma'am.
Okay, because there were one school of thoughts who said so.
Okay. What concentration of TTC are you using? Was used in the paper, I'm sorry.
I think it was only the 50% of reduction and that
0.05% TTC use किए।
0.05%?
Yes, ma'am.
Okay.
Okay, so what would you think about the cut vegetables and the product if you have to extend this particular work for the frozen cut plant samples? What do you do? Would you derive the information or the inference as?
Ma'am, actually my question समझा नहीं proper, इसका blurring हुआ।
So you say that, okay, fine. You say that freezing leads to the deactivation of dehydrogenase, right?
Yes, ma'am.
So
no, ma'am. Actually, in this paper, not due to dehydrogenase. There is something other enzymes which is responsible for inhibition of a synthesis of a substrate and cofactor.
Go to your results slides, beta. Result and conclusion के slide पे जाओ. रुको 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 आगे.
Ma'am, this
freezing and subsequent addition of specific substrates and cofactor make possible the demonstration of specific dehydrogenases. Right, the dehydrogenases enzymes do have a play in this freezing of cutting of the plants, Kalam.
Yes, ma'am.
Okay, so we can discuss it later on, but please remember that. Okay.
Yes, ma'am.
Okay. Yeah, we can go to the next participant.
Yes, can we have the next one then?
Yeah, yeah.
Okay.
Yes, ma'am.
Yeah, sure. Maksud Ali, you're the next one.
Yes, ma'am. Yes, ma'am.
I have made you the co-host already. After Maksud, it is Shoaib.
Yes, ma'am.
Yeah, Shoaib, you also be ready. And students, one request, please stick to the time limit.
Good evening, ma'am. Good evening, everyone. Am I audible?
Yes.
Yes, yes.
This is my topic for presentation, that is colorimetric determination of alpha tocopherol, that is vitamin E, by Emery and Angel, received November 5th, 1938. These are the contents under which we will discuss about the introduction, experimental part, standard cut for carotene, determination of tocopherol in oils and concentrates. Introduction. Investigation of Evans and Carrer have shown that tocopherol possesses reducing properties. According to Carrer, ferric chloride is not suitable for potassium titration which are formed during the reduction of ferric chloride are immediately removed by the dipyridine, so that there is a considerable difference between the result obtained by oxidation with ferric chloride alone and in the presence of dipyridine. Embry and Angel demonstrated this fact while oxidizing carotene with ferric chloride. In the presence of dipyridyl, carotene is oxidized to a colorless compound, whereas in the absence of dipyridyl, the carotene is oxidized incompletely. Embry and Angel have determined tocopherol in different oils like saponified and unsaponified oil. Experimental part, solution and reaction. Here the solution are pure ferric chloride in ethanol, dipyridyl, benzene. Here the reaction is carried out in alcoholic solution, but the presence of benzene in the reaction mixture has no influence on the color. We use benzene to dissolve unsaponified oils because they are sparingly soluble in ethanol. Next is 0.1 to 4.4 mg tocopherol. Procedure, to 1 ml solution containing 0.1 to 4.4 mg of tocopherol are added 1 ml of ferric chloride solution, and after mixing 1 ml of dipyridine solution, the volume is made up of 25 ml, and red color of the ferrous dipyridine complex is measured after 10 to 15 minutes with a photometer. Blank with ferric chloride and dipyridyl is also prepared and is placed in the other cell of the photometer. Standard cut of carotene. As carotene also reduces ferric chloride, large concentration of carotene may also cause difficulties and must be taken into account. Here the figure gives the standard cut for beta using the part which applies the small quantities of carotene, because the quantity of carotene in there more than 0.01 mg, so they calculated a conversion factor of 1. Determination of tocopherol in oils. Saponification. 5 grams of oil are saponified with 10 ml of 2N-methyl alcoholic potassium hydroxide at boiling temperature during 10 minutes in an atmosphere of nitrogen. The mixture is diluted with 15 ml of methanol and 40 ml of water and extracted 3 times with 50 ml of pure ether. The combined ether extract are washed with water with dilute potassium hydroxide solution and again with water until alkali free. The extract is dried over calcium chloride operated in vacuum in a carbon dioxide atmosphere and finally taken up in a suitable volume of ethanol. Thank you.
How has this paper helped science, bacha?
Sorry, ma'am. Can you repeat, ma'am?
How has this paper or how has this research helped science at large?
Ma'am, we can find out vitamin E from different oil. Like today we are seeing ads of different oil company brand of oil company. So we can find they mention they mention their product. They mention in their product vitamin E content. So we can find out the vitamin E from different oil.
But it was not colorimetric estimation, na?
And you said you have.
Hello. Hello.
Yeah, it was. Was it a colorimetric estimation, Maqsood?
Yes, ma'am. This is colorimetric colorimetric method, ma'am.
You said it is saponification, right?
Yes, ma'am. No, we are using, ma'am, oil with we are using saponified oil, ma'am.
Yeah, can you go back to that particular slide where you had written saponify the process? Ma'am was asking you about the significance, right? So when you say that,
Yes, ma'am.
so can you go back to that saponification wala? Right.
Yes,
so what
ma'am.
is
This
this?
one.
Yes. So this is 2n is 2 times n or 2 normality ka n जो by mistake n small n हो गया?
No, ma'am. It is a 2 and it is 2 normal n, ma'am.
अच्छा, तो वो capital रहना चाहिए ना मतलब?
Yes, ma'am.
Okay, fine, right? So colorimetric estimation अगर इसमें है तो कौन से नैनोमीटर पे लिया तुमने?
Ma'am, 560 nanometer, ma'am.
तो वो स्टैंडर्ड के लिए लिया ना तूने?
Yes, ma'am.
मतलब उन्होंने स्टैंडर्ड के लिए लिया ना, तो मैम क्या पूछ रही है ये स्टडी के हिस्से how was this particular paper significant for the other studies?
Other studies, ma'am. Ma'am, मैम हम लोग ना इससे ना मैम अलग अलग सैंपल से भी फाइंड आउट कर सकते हैं विटामिन ई, मैम, बिकॉज सेम रिएक्शन देगा वो ट्राइपैरिडिल जो है ना मैम, वो ना क्या वो फेरस आयरन के साथ मिलके सेम रिएक्शन रेड कलर कॉम्प्लेक्स देगा, तो वी कैन फाइंड आउट विटामिन ई फ्रॉम ब्लड आल्सो। अभी ब्लड में पता करना है कि है कि नहीं है, तो हम लोग वी कैन फाइंड आउट बिकॉज इट विल गिव सेम कलर दैट इज रेड कॉम्प्लेक्स।
Fine, that's okay.
Okay, madam, then should we move further or you have further questions for me?
No.
Next participant is Akhtar Sheikh.
Yes, ma'am.
Yeah. Akhtar, I have already made you co-host.
Chanvi ma'am, next is Maqsood, right?
Maqsood is, we have just
Maqsood
completed Maqsood. Maqsood has completed his presentation.
Yeah, no, now coming one is Akhtar.
This is Shoaib Akhtar. Shoaib Akhtar Sheikh. Okay, am I correct? Shoaib Akhtar Sheikh?
Yeah, yes, ma'am. Yes, ma'am.
Yeah. Excellent.
Ma'am, is my am I audible?
Yeah,
Yes,
you're
yes, you
audible.
are.
Your screen is also visible.
So yeah, good evening, everyone. Today I'll be talking about the research paper which tells us about phytochelatins. So the topic is induction of heavy metal binding phytochelatins by inoculation of cell cultures in standard media. The paper was given by E. Grill, J. Thuman, Lineker, and M. H. Zink, which was received on July 25, 1988. Here's the original paper. So first of all, let's talk about phytochelatins. What are phytochelatins? Phytochelatins are oligomers of glutathione produced by the enzyme phytogelatin synthase. Phytochelatins act as chelators and are important for heavy metal detoxification. These peptides consist of repetitive gamma glutamyl cysteine units with a carboxyl terminal glycine, and they range from 5 to 17 amino acids in length. Here's the general structure of a phytogelatin. Phytogelatins are assumed to be involved in the accumulation, detoxification, and metabolism of metal ions such as cadmium, zinc, copper, lead, and mercury in plant cells. The general structure of this peptides is glutamine, cysteine, and glycine. Uh, uh, they range from 2 to 11 in amino acid length. Here's the introduction of the paper. A large increase in phytogelatin synthesis occurred when cell cultures were transferred, where cell cultures of different plant materials were transferred from spent medium to fresh standard media. A spent media is the one which has been already used for growth of a for growth of cell culture, and fresh standard media is the freshly prepared one. Inoculation of plant suspension cells from logarithmic or stationary growth phases into fresh cell culture media causes considerable changes in the metabolism of the cultured cell. For instance, the activation of protein synthesis, specific increase of enzymes of phenyl propanol pathway, etc. The function of phytochelatins were assumed to be detoxification and homeostasis of heavy metal ions in plant. These ions interfere with the sulfhydryl groups of protein. Therefore, these reactive metals should be physiologically inactivated in the cell to allow undisturbed metabolism in plants. The concentration of these heavy metal ions is regulated by the synthesis of phytochelatins. Here's the plant material and methodology. The plant material used were cell cultures were grown in a 75 ml flask, 300 ml flask, which consisted of 75 ml medium on a guided shaker, which whose RPM was 100 at 23 degrees Celsius in continuous light. The intensity of light was 650 lux, and inoculation into fresh medium was performed at late stationary phase. Media. The media used for the growth of cell cultures was a street media or the Linsware and Scoop media. In the experiments indicated cells were grown in the same media but omitting both copper and zinc salts. The assay for phytochelatins. Suspension cells were separated from media by suction filtration. Aliquots of 0.4 gram were suspended in a solution containing one normal NaOH and one normal sodium borohydride per ml. After sonication and centrifugation of the sample, the supernatant was acidified with 3.6 normal HCl. Precipitating material was sedimented again and the cleared extract was separated by HPLC. Detection of the sulfhydryl containing phytochelatins was performed by post-column derivatization with 74 micromolars, 2-nitrobenzoic acid, elements reagent in 50 millimolar potassium phosphate buffer at a flow rate of 2 2 ml per minute and detection at the 410 nanometers. Zinc and copper ions were detected by atomic absorption. The results and discussion. The first indication that phytochelatins were involved in heavy metal ion homeostasis was the observation that plant suspension cells grown in standard medium lacking zinc and copper ions contain no detectable or very low levels of phytochelatins. However, inoculation of these cells into the unmodified medium providing physiological concentration of essential heavy metal ions led to an immediate rise in phytochelatin concentration, which reached its maximum in mid-log to late log phase. Here's the graph showing the amount of phytochelatins present in the cell cultures of Rolfe serpentina. Look at here, we can see at the sixth day, the amount of phytochelatin present in the cell cultures was maximum, and later on it went on decreasing as the cells continued to grow. Rauwolfia serpentina cells are grown for one growth cycle in a copper and zinc-free medium to deplete phytochelatins, followed by transfer of cells to LS medium, that is the Linsware and Scoob medium, which resulted in 100-fold increase in total amount of metal chelating peptides on 6th day after inoculation. During stationary phase, a strong decrease of the total amount of metal chelating peptides in the cells was observed. Since no phytochelatins could be identified in the culture medium, this finding clearly indicates that phytochelatins are degraded, probably to their amino acid constituents. Thank you.
All right. So
No
sure,
questions.
sure.
Yes, ma'am.
This was detected by means of AA, right?
Sorry, ma'am, I didn't get you.
This was detected, the work was detected by using AA, isn't it?
AA.
Yeah.
Atomic absorption, yes, ma'am.
You only have mentioned it in your results and discussions, right?
Yes, ma'am. Actually, you
So bhul gaye.
no, you actually used the abbreviated form, so I didn't get it.
So, is there any other detection method which was better, or other which is better? Do you think there is other detection methods for this heavy metals using the same experimental lines?
Actually, I have got no idea, ma'am. Like, what other instrument can be used to find the microscopic, like the biomolecules? These are biomolecules, very small elements.
Right. It is not, I mean, a microscopic kind of phase. It is
Yes,
obviously
ma'am.
by means of instrumentation. Okay.
Yes,
So,
ma'am.
is there anything else which could have taken instead of Fina, or is there any specific reason जो R serpentina लिया है? ऐसे
No,
कुछ?
ma'am. Actually, they use different cell lines, but they specifically mentioned about Rauwolfia serpentina. They also mentioned that they took other cell cultures. But they didn't mention the any other names. They only told that they used Rauwolfia serpentina. While also they mentioned that other cells also showed the same kind of results.
Okay, so others मतलब कौन सा?
Ma'am, actually I've got no idea. They didn't mention in the paper.
They didn't mention in the paper.
No.
Okay, fine. Right. Yeah. Thank you.
Thank you, ma'am.
Sucheta madam, questions for him?
Has she logged off? Probably network issue. But she said no questions for him.
Okay,
Yeah.
fine. So the next one is Nahid.
Oh, yeah, okay.
I request all the students and the teachers, please sign the attendance link. I'll post it one more time.
Okay, so the next one is Nahid. What
Yeah,
about
the next one is
Karishma
Nahid.
and Rafia?
They are all there, but
Okay.
is Nahid here? Nahid, are you there?
Yes, ma'am.
Okay.
So Nahid, you can proceed with your presentation.
Okay, no problem.
After Nahid, Karishma, Rafia, and Irshad, you be ready.
Good morning, everyone. My topic for presentation is the B-type cyclin kinase inhibitor P40SIC1 controls the G1 to S transition in Saccharomyces cerevisiae. The contents are introduction, research hypothesis, theory, findings, conclusion, and references. Introduction. When yeast cells reach a critical size, they initiate bud formation, spindle pole body duplication, and DNA replication almost simultaneously. All these three events depends on activation of CDC28 protein kinase by the G1 cyclin CLN1, 2, and 3. They showed that DNA replication also requires activation of CDC28 by B-type cyclin CLB cyclins. A sextupule CLB1 to 6 mutant arrests as multi-budded G1 cells that resemble cells lacking the CDC34 ubiquitin conjugating enzymes. CDC34 mutants cannot enter S phase because they fail to destroy P40SIC1, which is a potent inhibitor of CLB but not CLN forms of the CDC28 kinase. In wild-type cells, P40SIC1 protein appears at the end of mitosis and disappears shortly before S phase. Proteolysis of a cyclin-specific inhibitor of CDC28 is therefore an essential aspect of the G1 to S phase transition. Next is research hypothesis. CDC28 associates with CLN cyclins and 6 B-type cyclins called CLB6. CLB1 and 2 proteins appear during G2, whereas CLB3 and 4 appears at the end of S phase. Mutants lacking CLB4 enters S phase with normal kinetics but fails to form bipolar spindles. Transcripts from CLB5 and CLB6 genes appears in late G1 around the same time as those from CLN1 and 2. It is possible that a rise in CDC28 kinase activity associated with CLB5 and CLB6 might be more directly involved in activating DNA replication than increases in CLN1 and 2 kinases. Because S phase but not budding is delayed by at least 30 minutes in CLB5 and CLB6 double mutants. There are two problems with this hypothesis. First one is S phase is only delayed in the absence of CLB5 and CLB6 cyclins. And second one is premature CLB5 gene expression from the GAL1 promoter doesn't advance the onset of S phase. Next is theory. CLB5 CDC28 kinase activity peaks around S phase. CLB5 appears as cells begin to bud at 90 minutes, preceding the appearance of the G2 specific CLB2 cyclin. CLB5 associated kinase activity appeared as cell entered S phase, and the absence of CLB5 in the starting population of G1 cells suggests that most, if not all, CLB5 protein made during late G1 and S phases is destroyed by the end of cell division. Next is B-type cyclins are required for DNA replication. CLB5 gene is under the control of GAL1 promoter, and the strains can only grow in medium containing galactose at 25 degrees Celsius. Inactivation of CLB1 to 4 doesn't affect progression through S phase, suggesting that these mitotic cyclins are not normally needed for DNA replication. Finally, CLB5 alone can trigger that seems to be a complete S phase in the absence of CLB1, 2, 3, 4, and 6. It has been suggested that in some strains, CLB1 to 4 are needed to complete S phase. Third is post-translational control of CLB5 kinase in early G1. They concluded that CLB5 CDC28 kinase is negatively regulated in early G1 by a post-translational mechanism, which would explain why premature CLB5 expression fails to advance S phase. Next is CLB CDC28 kinases are inactive in CDC4, CDC34, and CDC53 mutants. The absence of CLB2-CDC28 or CLB5-CDC28 kinase activity in CDC34 mutants contrast with the high level of kinase associated with CLN cyclins in these mutants. Next is G1 cells contain an inhibitor of the CLB5 kinase. The G1 cells, but not those from other cell cycle stages, contains an inhibitor of CLB-CDC28 kinases, which could be responsible for the lack of CLB5 kinase in the CDC34 class of mutants and in early G1 cells expressing CLB5 protein. Then P40SIC1 binds to and inhibits the CLB5-CDC28 kinase. P40 is encoded by the SIC1 gene and is present in G1 cells that contain the CLB5 kinase inhibitor. Deletion of SIC1 is not lethal, which enabled us to test whether the inhibitory activity of wild-type G1 or CDC34 cells depends on P40. Inhibition exerted by both types of extract is abolished in SIC1 mutants. Thus, P40SIC1 alone could be responsible for the CLB5 kinase inhibitory activity present in G1 and CDC34 extracts. Then disappearance of P40 at the G1 and S boundary depends on CDC34. SIC1 dependent inhibitory activity is high in G1 cells but undetectable in S, G2 and M phase cells. Then effects of deleting SIC1 on S phase entry. SIC1 is might be only one of the several factors that prevents premature DNA replication in wild type cells. They also notice that SIC1 cells take longer than wild type to complete S phase. In wild type, the distribution of DNA contents become bimodal very soon after S phase entry, whereas in the mutant, it drifts more slowly to the to an position as a single peak. Then findings. Activation of the CDC28 protein kinase by the G1 specific cyclin 1, 2, and 3 cyclins is required for budding, DNA replication, and spindle whole body duplication. 3 additional genes CDC4, CDC34, and CDC53 are more specifically required for S phase entry. CLB kinases are necessary for initiation of DNA replication. Mutants that lack all 6 CLB B-type cyclin activities form buds but fail altogether to enter S phase, despite having active CLNs. Whereas CLNS means G1 cyclin cannot trigger S phase in the absence of CLBs. Ectopic expression of either CLB5 or CLB PADB can trigger S phase in the absence of G1 cyclins. Thus, CLB CDC28 kinases and not CLN CDC28 kinases may be the true S phase inducer. Then second one is destruction of P40 SIC1 is needed for CLB kinase activation and S phase entry. 3 different mechanisms prevent CLB kinases from becoming active in early G1. First, CLB genes are not transcribed until late G1, S or G2. Second, CLB cyclins are degraded as cells exit mitosis and at least in the case of CLB2 remain unstable during the following pre-start G1 period. They discovered that a third mechanism, CLB CD28 kinases are kept inactive by an inhibitory protein P40SIC1 that accumulates as cell exit from mitosis and only disappears shortly before S phase by a process that depends on the CDC34 ubiquitin conjugating enzyme. Third one is regulation of P40SIC1 levels. SIC1 is transcribed throughout the cell cycle with a peak around cell division, but P40 disappears at the G1 to S transition and doesn't reappear until cell division. It is not known what turns P40 degradation off and thereby triggers accumulation of P40 at the end of mitosis. It occurs around the same time as the onset of CLB2 proteinases and may contribute to contribute to the inactivation of CLB kinase, which is to be necessary for exit from mitosis. The reappearance of P40 could explain why cells can exit from mitosis in the presence of moderate amount of a non-destructible CLB2 protein. And fourth is functions of P40SIC1. Cells lacking P40 are viable but have an increased frequency of broken and lost chromosomes. They help to prevent premature entry into S, and it may thereby prevent error in DNA replication. It also
Please stick to your time.
Okay, ma'am. It also seems important for the rapid completion of S phase. P40 may actually help CLB kinases to function even though it inhibits them. Then regulating S and M phases. Phosphorylation of P40 by CLN1, CDC28 and CLN2, CDC28 kinases might trigger its degradation by the ubiquitin conjugating enzyme CDC34. This could be relevant to the mechanism by which replication during G2 is avoided. Then conclusion. Results indicate that proteolysis is a key mechanism for regulating the G1 to S transition in yeast. It is likely that degradation of mitotic cyclins are required for exit from mitosis, and it is possible that degradation of other proteins might be required for cystochromatid segregation. Because of its irreversibility, proteolysis may be an ideal mechanism for ensuring the precision of cell cycle events. These are my references. Thank you.
Which paper was this? Nahid?
B-type cyclin kinase inhibitor, P40SIC1.
No, in which journal has it been published?
Dr. Bogras 7.
And how is it? How is this finding helped science as such? Give me just खाली बताओ कि ये फाइंडिंग करने के बाद क्या कंक्लूड हुआ है.
मैम ये फाइंड आउट करने के बाद ये पता चला है कि जीवन से जीवन टू एस फेज ट्रांजिशन जो है वो P40 प्रोटीन जो है उसके इनहिबिशन की वजह से होता है.
What does G and S stand for in cell cycle?
Gap 1 phase and synthesis phase.
Okay.
Okay, Nahir.
Yes, ma'am.
If there is some mutation occurring at that particular level, does it go to the further phase in the cell cycle, or it is restricted to S?
Ma'am, it will be restricted to S phase.
Good, right. Okay, yeah, that's it.
Can we have our next participant then?
Yes.
Yeah. So next participant is Karishma Gupta.
Yes.
After Karishma, it is Raphael Sheikh. So Raphael, you also be ready. Karishma, you can start with your presentation.
Janvi ma'am, how many of them are left now?
Madam, they're quite
Big listed.
Yeah. I'll just tell you, ma'am, how many are left.
Yeah.
5 are left, madam.
Okay, fine. Now, students, please stick to the time limit given. Okay, and see, more important that we are looking out is what is your understanding about the paper. Okay, and how is this finding helping science? Okay, so please stress on those parts rather than going into the actual material and method. It's okay, but right now since we are less of time, okay, let us concentrate on these two aspects currently. Okay?
Okay, students, please follow whatever madam has given instructions. Please follow those. Okay, we can start with Karishma. Please start. करिश्मा आपका ऑडियो बंद है।
शेयर ऑडियो ऑन करो।
Yes, ma'am. Am I audible?
Yes, yes.
Yes.
Can I start, ma'am?
Yes, yes. Please start.
Good morning, everyone. My name is Karishma Gupta. And today I'm going to present paper titled as Control of Gene Activities in Palatine Chromosomes of Karishma Gupta. Title written by Marcus Lazid and Lawrence I. Gilbert in 1969. Let us see the abstract. Injection of ecdysone into chironomid larva gives specific gene stimulation, which increases the RNA synthesis. They show gene activities and
Karishma, could you increase your volume? We are not able to hear.
Okay, ma'am. I'm audible, ma'am?
Yeah.
Just a minute, ma'am. Shall I start, ma'am?
Yes.
Sorry, ma'am.
No problem. Please start.
Yeah. Let us see the abstract. Injection of ecdysone into chironomid larva, which gives specific gene stimulation, which increases the RNA synthesis. It shows that. Sorry, ma'am.
Is the presentation visible?
No, the presentation is not visible.
Karishma, बेटा share करो।
एक मिनट मैम।
Don't panic.
No मैम, एक्चुअली मैंने शेयर किया है।
वो शेयर साउंड के समय फिर से तुमने शेयर निकाला ना, तो अभी वो नहीं दिख रहा है।
हाँ मैम, सॉरी।
Yeah, no problem. Yes.
Is it visible, ma'am?
It will be visible now. Yes. Yes.
Let us see the abstract. Injection of ecdysone into chironomid larva, which gives gene-specific stimulation, that is RNA synthesis. In this paper, they show the gene activities in this and formation of puff. They uses two techniques, which is cytological technique and autoradiographic analysis of chironomid tendons. It reveals that it stimulates Balbany ring 1. Balbany ring are tissue-specific and account for significant percentage of non-nuclear region. Application of juvenile hormone result in decreased activity of Balbany ring 1, suggesting that they two hormones may act oppositively. Ecdysone, which increases the RNA synthesis, and juvenile, which decreases the RNA synthesis. Let us see the introduction. In introduction, Palatine chromosomes are very useful for eukaryotic interphase. It shows as genome as whole. According to the Clever and Carlson, the molting hormones ecdysone can influence the activity of two specific loci on Palatine chromosomes. In this paper, they choose two specific loci, which is I18C and 1918 I19C. Although ecdysone induces molting in insect and juvenile hormones, titer determines the nature of molt. Present paper describes the effect of juvenile hormone on chromosomes puffing. Now, what is Palatine chromosomes or giant chromosome? Giant chromosomes or Palatine chromosomes are oversized chromosomes which commonly found in salivary gland of larval stage of Chironomys tendens. Band of Palatine chromosomes at certain region become enlarged to form swelling are called puff. The swelling represent unfolding of individual chromosomes in the band. The puff contain large amount of RNA, which is mainly in the form of mRNA. Now what they use for their materials and what they follow the methods. They choose animal and they treated it with ecdysone treatment and juvenile treatment. In animal, they choose fourth instar larvae were staged according to the method of Grieger. Young larvae have little or no endogenous ecdysone, while old pupa has critical hemolymph titer. During ecdysone treatment, synthetic ecdysone was dissolved in physiological saline and young larva was injected with 0.5 to 0.7 mu. In juvenile treatment, several substances such as DL juvenile hormones, etc. Possessing juvenile hormone. Sorry, sir.
Is there any disturbance? When is it?
Karishma, you continue, please. It's not
Okay, ma'am. These substances were either diluted in olive oil or injected in old pupa, which is they can use both the method. First, they can use it by injecting in old pupa, or second, they can apply it externally. In this research paper, they shows two techniques. First is psychological technique and second is autoradiography. In psychological technique, after 2 to 4 hours, treatment of salivary gland fixed, stained with acetic origin and squeezed. As you can see such a beautiful picture in which they choose fourth young instar larva. The left side A is control, which is exactly normal, and the second is injected with ecdysone, which is shows the Balvenia ring 1, 2, 3 are beautifully indicated in this diagram. In autoradiographic technique, after 4 hours, salivary gland were removed and autoradiographic technique prepared. Examined for H-uridine incorporation. The each Balvenia rings blackened areas are densely labeled. As you can see that the A, which is normal, and B, it's demonstrating the H-uridine incorporation into Balvenia ring and Balvenia ring 1, 2, 3. It shows in this phase clearly. Let us see the observation. In observation table number 1, it's revealed that there is a definite increase in puff frequency at the certain region, like I18C and IV2B, and it decreases at the region 119A. Although the activity of BR3 is also enhanced by ecdysone, the absolute increase is much less. As you can see the table number 2, effect of ecdysone H-uridine incorporation into Balvenia ring, it shows that the IV2V of 5 to B, which is shows in increasing, and RVI1, it is slightly decreases with RVI1. In third table, it shows the effect of substance with juvenile hormone activity in chromosomes morphology. In table number 3, it shows the puff is formed at the region I19A, while puff I18C are reduced. This sequence, first, second, third table, it's the sequence of low activity, high activity, and low activity, which is unaccepted by researchers. In fourth table, the effect of synthetic mixture of Balvenia ring 1 morphology, it shows that the stage 2 and stage 3, which is decreased, and it shows that while increasing the activity of region I19A, it's reduced. And it shows that I18C, I19A, not active in pupal region, pupal mold. And what is the discussion in this research paper? Clever was unable to relate the activity of BRI1 to concentration of ecdysone in hemolymph either during normal development or after injection ecdysone. Our autoradiographic studies demonstrate that Balvenia ring 1 incorporate the greatest amount of H-uridine, which is maximally expanded and therefore considered as the most active stage. H-uridine incorporate into an insoluble material, which are used in these experiments. And short-term of juvenile treatment of young larvae causes induction of puff I19A, and this puff reduce short-term of treatment of ecdysone. If we treat ecdysone for long term, it take maximally 12 hours and it reappears again if we treat for long term. The exact mechanism by which ecdysone and juvenile hormone activate the specific chromosomes region described previously are not known with certainty. In this paper, it is shown. The apparent relationship between hormone titer and the activity of BR1 was experimentally produced treating last instar larva with ecdysone or possessing substance juvenile hormone activity. Thank you. And these are my references which I taken for the research paper. Thank you. Yes, ma'am.
Yeah, Karishma,
Yes, ma'am.
have you ever separated the have you ever seen the polyethylene chromosome?
Yes, ma'am.
Have you done that experiment in your practicals?
Actually, it is in my syllabus, ma'am, but because online practical, I didn't see it, but
So,
I studied about it.
okay, so that is the reason why you chose, or was there any other reason why you chose this particular?
No, ma'am. Actually, it is a very interesting topic.
Achha.
First reason that it is in my previous syllabus as well, and second, it is something new. We are normally see the cell division, but we doesn't see ever long chromosomes.
Okay, fine.
It is very beautiful experiment. That's why I like it and I choose it.
Achha, okay. So do you know difference between Lamprecht chromosome and polyethylene chromosome?
Yes, ma'am. The very simple is chromosomes are found in salamander, of egg cell of salamanders, and this chromosomes which is found in larva, bloodworm.
Okay.
Adenomous larva, or simple language is bloodworm.
Okay, fine, right. Yeah. So that's all from me.
Yes, ma'am.
Yeah, no question.
Thank you, ma'am.
Okay, can we have our next participant? That is Raphael Sheikh.
Okay, it's Raphael. Okay.
Yes, ma'am.
Raphael, you can start with your presentation.
Ma'am, is it visible?
Yes, it is visible. After Raphael, I think it is Irshad. Irshad, you also be ready.
Yes, ma'am.
Good afternoon, everyone. I am Raphael Sheikh, SYBSc student. My topic of presentation is on individual differences in human blood, which was discovered by an Austrian biologist, Karl Landsteiner, and his assistant and pathologist, Philip Levine. It is a common saying that same blood flows in everyone's veins. However, it was soon proved inapt by Landsteiner, who discovered various types of blood and won the Nobel Prize of 1930 in physiology or medicine for his discovery of human blood group. Out of those various discoveries of blood types, one is M, N, and P done by Landsteiner and Levine. Here is a picture of their research paper, which was published in the year 1928. Now, the common terminology used frequently will be agglutinogen, meaning antigens which are present on the surface of RBCs, and agglutinin, meaning antibodies which are present in plasma. Now, in this paper, they mentioned the findings of well-defined serological differences. Their test showed presence of an agglutinable property unrelated to the isoagglutinogens A and B, which was discovered previously. Now, the existence of individual differences in human blood was shown long ago by the means of normalized agglutinins, which was discussed A and B. Now, soon thereafter, attempts were also made to discover further differences in human blood in addition to group distinction. So they started performing experiments. They performed experiments using immune sera in dilution of 5 volumes of 5.4% glucose solution, 2 volumes of 3.8% sodium citrate solution for 3 volumes of blood. So the ratio was 5:2:3. They performed numerous experiments under different categories, out of which are reaction of inactivated immune sera with different types of blood, testing of immune sera for M, N, and P agglutinins, determination of frequency of agglutinins, and comparative study which was done on mammalian blood. Now, the very first experiment was performed using stock anti-human blood immune sera from rabbits. It was observed that the first 41 sera were exhausted with one sample of human blood of type O. And then it was observed that out of 41 sera, 4 contained agglutinins acting on a majority of blood, while other blood was not agglutinated. These tests showed the existence of an agglutinable property unrelated to the isoagglutinogen A and B, and there was not found a corresponding isoagglutinin in human serum. Later, efforts were made to produce such immune sera possessing these peculiar properties designated as M and were injected in rabbits. It was found that few of the rabbits produced potent specific sera for M, and some immune sera exhibited a different effect when they were absorbed with blood type M, and thus revealing the second agglutinable factor N and later P. Here are the results of experiment performed under reaction of inactivated immune sera with different blood types. Here, TR indicates trace amount of clumps seen. Single positive sign indicates clumps seen with hand lines or clumps of medium sign. Double positive sign indicates large clumps seen with naked eye. And triple positive sign indicates complete agglutination. Later, the frequency of agglutinins were determined. This is the table of frequency of M with 4 different blood groups. And this is the table of frequency of N with 4 different blood groups. Agglutination of immune sera M, N, and P with different blood types can be seen here. This is the picture taken from original paper. Here we can see agglutinations in some blood. This is how agglutination looks with blood type A and B. Comparative study on mammalian blood was done using numerous animals like macaques, rhesus, vervets, baboon, lemur, horse, cattle, sheep, rats, dog. N number of animals were used for this type of study. And the conclusion made was every individual human have its own characteristic serological feature. And this means that each and every individual having different plasma features and different signature features. There exist several agglutinable factors for which no agglutinins were demonstrated in human sera. That means the findings which were dealt in this paper thus have no direct bearing on the selection of donor in transfusion. That is, the MNP blood types doesn't interfere in transfusion. In transfusion, only Rh factor and ABO blood system is to be considered. Now, it is a revolutionary discovery made by Landsteiner and hence made blood transfusion possible, which has till date saved billions of lives. Now, imagine a situation where a person living in Mumbai with rare blood type requires a blood, and blood is to be bought from Thane. Now, time for blood clotting is around 8 to 15 minutes, and distance between Mumbai to Thane is around half an hour. So what if the blood which is to be transfused starts clotting midway? Here comes Richard Levinson, who discovered best anticoagulant by revealing the optimum concentration of sodium citrate. This prevents blood from clotting. And this discovery, M, N, and P blood group system, this opened the gateway of possibilities of different blood groups present in humans. Now, if blood group ABO system was discovered, we would have been thinking that this is the only blood group. But later, this discovery opened that yes, there could be further more blood groups present. Until date, we are known with 33 blood group systems. Here are some other significant discoveries of blood groups. Here are the references and image courtesy. Thank you so much.
Okay.
Yeah.
Currently, which blood group system are we following?
We are following MN and P blood group system.
And do you have an idea what is Bombay blood group?
Yes, it is the Bombay rare blood group, which is found 4 in a million, and it was discovered in Came Hospital. It lacks H antigen in the RBC, surface of RBC.
Right. So, Raphael, do you know what any significance of this in the world of forensic? Blacksters work?
Yes, it is also used for paternity testing, as if we find a blood, so we can eliminate those others who are not the father of that child. And also in crime scenes, if we find a blood which is having a blood group A positive, so we can neglect all those having all other blood groups, and we can concentrate on the people having A positive blood group. So it is also used in forensic and science.
All right. And anything related with polio? This might be
Actually, Landsteiner has done research related to viruses, but I've not read that.
Yours is the main thing related to the blood groups. That's the reason I said it is a very specific question. But yeah, that's okay. Thanks. Yeah, that's all from me. Actually. Nice.
Thank you so much.
Thank you.
Thank you so much, ma'am.
Okay. So can we have our next participant? Irshad, you are ready with your presentation?
Yes, ma'am. And just give me a second.
Yeah.
And then we have Simran and Ansh, right?
Ma'am, now is my screen visible?
Yes, your screen is visible.
Okay.
Next is Simran Kathivas. Simran, you also be ready. Simran, and last participant is Ansh. Both of you be ready.
Should I start?
Yes, Irshad, you can start.
Good evening, everyone. I'm Mohammad Irshad Khan from TYBC Zoology. I'm going to present you one of the groundbreaking experiments in the history of life sciences, the Miller-Ure experiments, which is titled as a production of amino acids under primitive Earth conditions. These experiments was conducted by Stanley L. Miller and Arnold C. Ure. The Miller-Ure experiments, or Ure-Miller experiment, was conducted to know the origin of life in the primitive Earth conditions. This experiment was performed in the year 1952 and published in the year 1953. The experiments have provided important breakthrough in the study of origin of life. It has confirmed that several key molecules of life like amino acids and hydrocarbons could have been synthesized on the primitive Earths. These simple molecules would then have been able to take part in the prebiotic or complex life processes, leading to the origin of life. So what was this Ure-Miller experiment? Working with his Professor Arnold Ure, Miller designed an apparatus to stimulate the ancient primitive conditions of Earth. Like if we can't study a fish in an aquarium, if we can't study fish in a sea, set up an aquarium. So they have put up a flask half filled with water to model the oceans. This was getting boiled to mimic evaporation. This is then condensed, connected to other flasks where for gases of the atmosphere they choose methane, ammonia, and hydrogen in the ratio of 2:1:2. They had thought that this was the abundant gases on ancient Earth. They added a condenser to cool the vapors, allowing them to form droplets and fall back into the oceans like rain. Here, it gets collected in a U-shaped tube. In the ancient Earth, would have many sources of energy like sunlight, geothermal heat, and also thunder. So they have added 2 electrodes to the atmospheric flask for spark to provide energy. The contents of the flask was then treated with mercury chloride to kill any living microbes present in the apparatus. In vacuum evaporation was also used to remove amines by concentrated sulfuric acids. Also, they use barium hydroxides to remove the ampholytes. As the temperature in the primitive earth is reducing, they have removed oxygen by using the vacuum pumps. After one day, what they have observed is the liquid present in a U-shaped tube changes its colors to noticeable pink color. And after the running these experiments for a week, it has changes to red color and turbid. Finally, they synthesize this mixture by paper chromatographic methods. For paper chromatography method, they have used two-dimensional paper chromatography in which first solvent system is butanol, acetic acid and water, and the second solvent system is phenol and water. Ninhydrin is used as a spraying reagent. As you can observe here, it is a picture from our original paper. We can see here some spots on the chromatographic that were dark and some are light spots. All these spots were amino acids which were identified on the basis of their RF value, the colors and size of these spots. The amino acids that were identified were glycine, alpha and beta alanine, alpha amino butyric acids. There were other amino acids, but the spots were too weak, so we couldn't find out what was it. We can conclude that on the basis of this experiment, that origin of life on primitive Earth was formed from inorganic components. And the reducing atmosphere also provides energy in the form of electrons to the atmosphere to form more complex molecules from simple ones, so the life continues. At last, they have successfully completed these experiments in support of the Oparin and Haldane theory. And the solution which was present in the YouTube U-shaped tube is also get compared by the what was Oparin and Haldane told, like the Haldane soup, or we can say it as primordial soup, where in the oceans the crozovets were present. It also provides the theory that first life was also originated on water. And these were the credits from where I have took the information all. And at last, I would like to say thank you to each and everyone for being patient ears to me. Thank you all.
So which was the first biomolecule to be formed?
Ma'am, amino acids.
Have you ever separated the chromatogram that you showed? Did you do that ever during your practicals?
Yes, ma'am, I have done it.
Okay, so which are the amino acids that you used, or the proteins, or whatever?
Ma'am, at that time we have used chlorophylls.
Amino acid.
Ma'am, are you talking about mine experiments or this experiment?
Separation of amino acid using chromatography technique. What you
yes,
showed, right? The chromatogram that you showed?
Yes, ma'am.
Okay, so you have done with chlorophyll, but not with amino acid?
No, ma'am, we haven't used amino acids yet. We have done it with chlorophylls. We have find out different chlorophyll A, chlorophyll B, carotenes, and xanthophylls. We have done with what was it?
Right. So why did you choose this particular paper? Any reason for that?
मैम ये टॉपिक ना मैम मेरे को बहुत पसंद है एक्चुअली, क्योंकि ना ये सिर्फ मेरे को नहीं, ये सबको जानना है मैम कि फर्स्ट लाइफ कैसे आई थी अर्थ में. तो ये टॉपिक के बाद में मैंने इसलिए चूज किया था ना, क्योंकि मैं इसका खाली अपर पार्ट जानता था. ये फर्दर स्टडी पढ़ने के बाद ना मेरे को इसका बैकग्राउंड नॉलेज और भी काफी कुछ पता चला है, जिससे मैं जान पाया कि उन लोग ने, जैसे कि इन लोग ने क्रोमेटोग्राफी यूज किया था एंड अदर्स, कि ग्लास अपैरेटस यूज करने की वजह से इनका सोल्यूशन भी टर्बिड हो गया था. अगर वो कुछ और सब्सटेंस यूज करते तो शायद वो टर्बिड भी नहीं होता. एंड ये सब भी पता चला, इसकी वजह से मैंने मैम ये टॉपिक चूज किया था.
अच्छा, तो बाकी का भी पढ़ा तूने, क्या क्या
yes,
है? ओके, वेरी गुड, फाइन. ऑलराइट, दैट्स ऑल फ्रॉम मी.
Sucheta, madam.
Done, ma'am.
Yeah, Sucheta, madam.
Yeah. No problem. Thank you. Next.
Simran.
Can I share the PPT?
Yes. Students, I hope you all have signed the attendance sheet. And I request the staff members present, you also please sign the attendance sheet.
Am I audible, ma'am?
Yes, yes.
Janvi ma'am, even we are supposed to sign?
That sir has posted the message that everybody should sign the attendance sheet,
Okay.
including the staff members.
Yeah, we'll do it.
Madam, I can send you the link on your personal WhatsApp.
Yeah. Sure. Thank you so much.
Thank you.
Can I start, ma'am?
Yes, beta.
Yes.
Good evening, one and all. Myself, Simran Katwas from TYBSc Zoology. Today, I am going to present a research paper, and the title is Modernization of the BOD Test for Determining the Efficacy of Sewage Treatment Process by author Claire N. Swear and Leland Bradley. It was published in the year 1946. This topic belongs to a subject, applied component, which talks about environmental science and methods of improving or managing the waste and pollution cost. Basically, what is BOD and what is the significance of modern technique to test the BOD level of water? BOD tests indicate the amount of organic pollution present in an aquatic ecosystem. It is the important component for the environment management. The government has imposed certain laws in order to manage the released water. It is useful in designing of treatment plants and calculation of wastage load in water. In simple words, BOD is the measurement of dissolved oxygen that aerobic microorganisms use to break down the excess organic matter present in water. To get more clarification, let's take a look at a graph. It shows that due to disappearance of clean water, or rather we can say as the organic matter increases in water, there is a decrease in dissolved oxygen and increase in BOD level. And on the right side, it shows when the organic matter present in the water is balanced, BOD level decreases and dissolved oxygen level increases. To test the BOD level, 2 cases were taken. Because of the severe pollution, British and American investigators used dilution technique to test and track the BOD level of wastewater caused by humans. Let's take a look at a graph that interprets the BOD curve. BOD curve occurs in 2 general stages, carbonaceous stage and nitrogenous stage. Test period for dilution technique was taken 5 days at 20 degrees Celsius, and within 5 days, 65 to 70% of organic matter has decomposed by microorganisms. And for the first 10 days, the curve shows exponential growth rate of BOD. In this period, most of the organic carbon is converted to carbon dioxide. After 8 to 10 days, the curve takes a sudden jump. This jump is due to the exertion of oxygen demand by the microorganisms that decompose nitrogenous organics and convert this to stable nitrate. One might ask, why nitrogenous oxygen demand generally begins after 8 to 10 days? Because the nitrifying bacteria are usually present in relatively small numbers in untreated domestic wastewater. And fortunately, the reproductive rate at 20 degrees Celsius is such that the population do not become sufficiently large to exert an appreciable demand for oxygen until about 8 to 10 days. To overcome this disadvantage, operators who are fully aware of the BOD curve have tried to hold nitrification to a minimum in order to produce a more satisfactory BOD removal record. Another case was taken at Sioux Falls. The treatment plant at Sioux Falls used to treat both industrial and domestic waste. The industrial waste is given 2-stage filter treatment before being admitted to activated sludge. The domestic waste can be treated within the industrial waste on the secondary filter or can be sent directly to the aeration tank where it gets mixed with industrial sewage. For final treatment, to study the BOD removal accomplished in the aeration unit,
which showed tremendous day-to-day variation. In order to determine VOD level, dilution technique was used. Two dilution was set up on the final effluent of 10% and 5% mixture as shown in the table. The result obtained with two dilution mixture considerably gave higher VOD values. In order to get the another result, another dilution mixture of 10% and 5% was set up of domestic sewage sample, which normally yield at the close BOD value. At the end of the result, it was concluded that difficulty was not in the dilution water, but rather in the nature of the final effluent samples. This is the image of the Sioux Fall, and this is the image of sewage treatment plant at Sioux Fall. Due to marked reduction of BOD, two techniques were obtained, chlorination and pasteurization, and it was studied through series of samples. In order to eliminate the nitrifying flora that cause nitrification, two technique was considered. First is pasteurization, which is used in terms of temperature, and second is chlorination, which is chemical based. Pasteurization consisted of heating the sample at 80 degrees Celsius and immediately cooling to 20 degrees Celsius. After cooling, the sample were shaken in stoppered flasks to saturate them with oxygen. In pasteurization, two tests was run. First was on final effluent and the second was on different types of effluent sample. And on comparison between pasteurized and unpasteurized sample, it was concluded that the pasteurized sample showed the elimination of nitrifying flora. Next question was, what might be the optimum temperature for pasteurization? To know the optimum temperature for pasteurization, studies were made at 5 degrees Celsius intervals between 50 degrees Celsius to 80 degrees Celsius. And the results after the study showed nitrifying flora readily surrendered at 55 degrees Celsius, and 80 degrees Celsius temperature was chosen randomly. The next is chlorination method. It was reported that chlorination of final effluent from biochemical treatment plant frequently result in marked reduction of BOD. Therefore, a series of samples were subjected to BOD analysis using pasteurized chlorination and unpasteurized portion. After the result obtained, the chlorination and pasteurization showed identical BOD values. On the other hand, unpasteurized samples showed much higher BOD values. The BOD factors that the factors that affects the BOD rates are the nature of waste, the ability of microorganism to degrade the waste in water, and the temperature. The application of BOD on environment is the determination of BOD evaluates the amount of biodegradable organic material present in effluent and polluted waters. The BOD test reflects the amount of dissolved oxygen consumed by bacteria. Dissolved oxygen is essential for the life of aquatic fauna and flora, and the BOD test is a measure of the ecological impact that effluent water may have on the receiving water bodies. The sources that affects the BOD are dead fishes, agricultural runoff that are shown in the images, and the industrial effluents, domestic sewage, and woody debris. Then the last is the summary of the research paper. The biochemical oxygen demand of effluent waters from biological treatment processes may be largely due to nitrogenous oxidation. The technique used such as pasteurization or chlorination of such effluent water will arrest nitrification and allow carbonaceous BOD to be measured, provided a seeded dilution is used and excess chlorine has been destroyed. Flash pasteurization at 60 degrees Celsius to 80 degrees Celsius with subsequent cooling at 20 degrees Celsius has been found to be satisfactory procedure for destroying the activity of the nitrifying organism. The measurement of residual carbonaceous BOD without interference from nitrogenous BOD allows determination of plant efficiency in terms of plant loadings. And here are the references that I have used, and the images was taken from the Google. And thank you for your attention and interest.
All right. So this was Simran. Was this Simran, right?
Yes,
Okay. So Simran, how does this particular study, or how has this study helped you in your, you said your applied component you have studied, or during your regular practicals also you have done. So how has this particular research journal helped you to understand the concepts better?
Ma'am, actually, uh, because of this test, we have known the actual concept of testing the waters that has been polluted by industries and localities that usually pollute this water.
Okay, so what happens if the biological oxygen demand is more?
It is insufficient for our daily uses.
Okay, and what about the flora and the fauna? So for example, if it is the aquatic flora and fauna, so what happens to them if the BOD is more?
The BOD levels are high, it affects the flora and fauna of the aquatic life, or in some cases, the aquatic flora and fauna might die.
Why?
Because the level of dissolved oxygen used by flora and fauna has reduced.
Used by flora and fauna or available to them?
Means available to them.
Okay, yeah, that's it from me.
Do you know the permissible limit of BOD? Say if it's an industry, okay, and if they need to discharge their effluent into the natural water bodies, there are certain norms, right, that they have to meet. So what is the permissible limit? Because before they discharge any of their waste, they have to find out whether it is within the limits, okay, which has been given by the governing authorities. So do you have an idea what is the permissible limit of for BOD if effluent has to be discharged?
Yes, ma'am. It is between 2 to 8 mg per liter.
Okay, fine. Okay, that's it.
So, madam, should we
Yeah.
have the
Yes.
last
Yes.
participant?
Yeah.
Okay, so we have last participant. And please don't leave, participants. We will have a photo of the session to capture the memories.
Okay.
And for the record. Okay, Ansh Vishwakarma, please start.
Yes, ma'am. Yes. Ma'am, start करूं?
Yes, yes.
Good evening, one and all. Myself, Ansh Vishwakarma, third year BSc Zoology student. I am here to present a research paper by Alexander Fleming, sir. And the title of the paper is on the antibacterial action of a culture of a penicillium with special reference to their use in the isolation of bee influenzae. This was published in British Journal of Experimental Pathology in the year 1929. This concept of discovery of penicillin is related to bioprospecting and zoopharmacognosy in my syllabus. Let me tell you relevance of my paper. Nowadays, we get immediate cure from microbial infections and life-threatening diseases, but it was not easy hundreds years back with limited instrument, facilities, and sources. Now let us see what happening in Fleming Sir's laboratory to achieve this. While working with Staphylococcus variant, a number of culture plates were necessarily exposed to air so that they become contaminated with various microbes. One region of within the plate was transparent, indicating that Staphylococcus lysis. So to find out further, a small sample was subculture to find out the inhibitory bactericidal property of the mold. Now further characteristics of the mold were studied. With respect to their medium, pH, which has to be maintained, and the temperature. Is the antibacterial body elaborated by all mold? To find out it, Flemingser had used various microbes, fungi, and 8 different strains of penicillin. Culture fluid were tested for antibacterial substances up to 1 month. And it was found that only one strain of penicillium shows inhibitory substances. It was indication that something inhibit the growth of bacteria, and mold growth filtrate was termed as penicillin by Flemingser. Now let us see the methods for examining the culture of antibacterial substances. So basically here 2 different methods were used. So first one was agar plate method. Furocut was made in the agar plate. It was filled with a mixture of equal part of agar and penicillin broth. Later streaking of various microbes was done at right angle from the furo to the edge of the plate. And it was observed that proximal portion of the plate become transparent, indicating that bacteria lysis. And the next method was serial dilution method. It was done to know at what concentration this penicillin behave as bacteriolytic substance. In tubes, penicillin nutrient broth were implanted with same volume of bacterial suspension, and incubation was done. Inhibition power of penicillin was checked by opacity and translucent appearance of the tubes. Now let us see the properties of the antibacterial substances and parameters which affect it. So first is effect of heat. Heating does not reduce its inhibitory power, but autoclaving does. Effect of filtration. Penicillin was passed through Sitz filter to obtain a sterile penicillin. Solubility. It is soluble in water, weak saline solution, and insoluble in ether, chloroform. Rate of development of inhibitory substance in culture was done to know how much time it required to inhibit the bacteria growth. Inhibitory power of penicillin on the growth of bacteria. This is the molecular structure of penicillin. Now let us see the inhibitory power of penicillin, how it was done. So basically here table 2 and table 3 shows the extents to which various microbes, pathogenic, non-pathogenic, are inhibited by penicillin. Table 2 shows the inhibition by agar plate method, and table 3 shows the inhibitory power when diluted in nutrient broth. It is clear from the table that penicillin contains bacterioinhibitory substance, which is very active towards some microbes while not affecting the other. The rate of killing of Staphylococci by penicillin. Experiments were conducted to find out which category penicillin falls, and it was noted that it belonged to a group of slow-acting antiseptics. And the inhibitory power of Staphylococci to be inhibited by penicillin was after 4.5 hours. Later, toxicity of penicillin was tested on various experimental animals like mice, rabbit, and some man with microbial infections. Different concentration of penicillin was used, and no toxic effects were observed on any of the organism. The use of penicillin to demonstrate other bacterial inhibitions. Sputum was spread thickly on culture plate in Fritz medium. Then 5 to 6 drops of penicillin is spread over one half of it. After incubation, it was seen that half untreated with penicillin show growth of streptococci and pneumococci, and penicillin treated half shows only growth of gram-negative cocci in the culture plate. Now let us see how it is done. The isolation of B influenza.
Please stick to your timing.
Ma'am. Yes, ma'am. In human, it is difficult to isolate the pathogenic bacteria because it occurs in association with other bacteria which grow more in number and mask it. In such cases, the first microbe is insensitive to penicillin while other is sensitive to it. Pfeiffer bacillus occur in association with streptococci and other bacteria in respiratory tract. Here, influenza is sensitive while influenza is insensitive and while others are sensitive to penicillin. Figure showed in the slide had upper half showing bacteria growth and lower half showing inhibition done by penicillin, and only influenza is colonies are seen here. Dr. McLean, Fleming-Suss colleagues made series of culture from 25 nurses swab samples, and it was seen that that penicillin inhibit the growth of other bacteria and allow the growth of influenza in the culture plate. This method is very helpful to bacteriologists to isolate influenza easily, but Fleming sir had not isolated purified form of penicillin. It was Howard Florey and Ernest Chen and at Sir William Dunn School of Pathology at Oxford University who turned penicillin from a laboratory curiosity into a life-saving drug. Here are a list of references which I used to collect the data. Thank you everyone for patient listening. Done.
Madam, any questions for Ansh?
Ansh, which bacteria were you referring? Gram positive or gram negative? Do you have any idea?
Ma'am, it's gram negative.
Are you sure about that?
Ma'am, the bacteria which was inhibited is gram positive and which was inhibited is gram negative.
Can you repeat what you said?
Ma'am, the bacteria which was inhibited by penicillin is gram positive and while other is gram negative.
All right, fine.
Ansh, can you tell me to which class of antibiotic does penicillin belong?
Ma'am, slow acting antiseptics. I need to read,
Okay,
ma'am.
tell me the action of penicillin on this organism. How is this organism inhibited? All right. Okay, just tell me penicillin is obtained from which organism?
Ma'am, it's fungi, ma'am.
Which organism produces? Am I audible?
No.
Yes, madam, you are audible.
Yes.
Yeah. A basic question. Just tell me which organism will you use to produce penicillin?
Ma'am, fungi.
Okay.
Penicillium, ma'am.
हाँ, ठीक है. Fine. Thank you. Then we am done.
Okay, ma'am. Thank you so much. I request everyone to switch on their videos for the photograph. Ansh, you can please exit your screen. I think Devraj sir is also here with us. Kiran sir is also here with us. I request all the participants and the teachers to please switch on the video for the screenshot.
Why is it not happening?
Sucheta ma'am?
I'm trying to. 1 second.
Yeah.
It's just flatly refusing me. Doesn't want to see me in my avatar, you know, probably. नहीं हो रहा यार. 1 second. मेरा sunflower ही निकाल लो.
अभी सनसेट हो रहा है ना मैडम, तो उसका डायरेक्शन चेंज हो गया होगा.
हो गया, वो हो गया. No, I'm sorry, it's not happening, Janu. It's okay.
Okay ma'am, no problem.
फोटो तो डाल वरना.
थांब जरा माता. ते सगळं चेंज करत बसायला लागेल तो.
कर ग, शचीला सांग.
साची कुठे आहे?
अरे हो.
ते काम होती ऑफ करायला कॉल.
I'm sure the students want to see you as well, right? But students, very well done. Great job, and it is good exercise, very well thought, and I'm killing the time, Urmi.
Sorry?
I said
he's killing the time so that you can switch on your video.
I am trying to do it, but it's just not happening. I don't know why that is the case. It's okay. I am flower for today.
I've taken screenshot.
And I would definitely like to congratulate each and every participant. You all have done wonderful job, and also would like to congratulate the mentors. They've put in a lot of hard work. Everybody has done extremely well, and congratulations to one and all, and congratulations to the mentors also for giving that kind of moral boost to the student. And students have also, a few of them have done their references, done their readings extremely well. They've prepared for the topic very well.
Kiran sir, would you like to say something? Devraj sir, few words for the students, Kiran sir and Devraj sir. Kiran sir, you are not audible.
I have been seeing these presentations, and as pointed out by our judges, they are very methodically, systematically done. I like it that students have given as being so particular about references, even for the photographs as well as reading. So that's well done to all the students. And since I am here, I thank Sukita madam and Veena madam for participating in our event.
Oh, that's our pleasure. No need to thank anytime. Because that is a learning curve for me as well, and it's an enjoyable process, and the sessions were so interesting. So please don't thank anytime. So in fact, I would like to thank you all for giving this opportunity.
Kiran sir. Kiran sir, would you like to say something to the students?
Can you hear me now?
Yes, sir.
Okay. I also would like to thank Sujita madam and Meena madam for judging the session. And yes, whenever I came to this session, I mean, I also found presentations really very well made and very well prepared. So I congratulate all the students and keep reading such papers. It will definitely help.
Thank you, sir. Thank you for giving us this opportunity. Great experience for us.
Okay. On the behalf of RAC, I thank.
Yeah, any student who would want to talk, who would want to say, because I'm sure this is something different, you know, and this is absolutely exciting that, you know, something has been discovered and you learn the story about all the hurdles and how they surpassed. So, you know, you feel as if you are in their shoes. So do you want to say something? I'm sure in a minute or so, we would like to hear from you.
Students, someone, would you like to speak something?
If they want, otherwise we can carry on.
Yeah, we are done with the session. I officially declare that the session is over for session 2, biology, botany and zoology session. I would once again, on the behalf of RAC, thank the judges, Dr. Sucheta Golwankar and Dr. Veena Kekar. And I want to congratulate all the participants. And I take this opportunity to thank Devrat sir, who has meticulously planned this entire event. And I would also like to thank the coordinator of the Research Advisory Committee of Jhunjhunwala College and our Vice Principal, Dr. Kiran Kolwankar. So thank you very much. Should I end the session?
Yes.
Thank you very much, everyone, and all the best.
Thank you, ma'am.
Okay, bye, everyone.
Bye.
Bye-bye. Have a good day. Have a nice vacation, 1 day break.
Thank you, ma'am.